Welcome to one and all. We take an immense pleasure to invite
Akila, you can say something. Once on the book. YouTube on just check. Akila or Dheeraj can say something so that we can check the acoustics. Just long the
Anil?
Sir, I can hear you, sir.
Okay.
Sir, can you see me?
Yes, I can see you, no problem.
Okay, sir.
I also hear you.
Okay, fine.
Take care.
Thank you.
Dr. Jagdish? So get ready by 9:45 or so. So Abhinav and you also. Yeah. Both of you coordinate. Okay. Talk to him. So introduction of the speaker, you got the notes, biodata. Once the technical section starts.
One minute. Okay.
Very good morning, ladies and gentlemen. I am Padmini and I
Wait.
highlight
a very good morning, ladies and gentlemen.
వస్తుంది పక్కన ఉన్నది వస్తుంది మీకు వస్తుంది మాట్లాడండి
మీకు ఇన్పిస్తుంది మీదే Hello.
Yes, sir.
మీ రిప్లేస్మెంట్ సార్
A very good morning, Varun.
హలో 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 వచ్చిందా సార్
Yes, sir. Rangarao, sir.
థ్యాంక్ యూ వెరీ మచ్ థ్యాంక్ యూ వెరీ మచ్
ఇప్పుడు వీళ్ళది కదా అన్మ్యూట్ ఉండాలి వీళ్ళది.
అన్మ్యూట్ చేసాను అన్మ్యూట్ చేశాను.
వాళ్ళిద్దరిది అన్మ్యూట్ చేస్తే సరిపోతుంది. వాళ్ళిద్దరిది అన్మ్యూట్ చేసుకోండి. నేను అంటే మేము మ్యూట్ చేసాం.
A very good morning, Varun.
yes yes చదవండి
ఒకసారి ట్రైల్ చేయండి. ట్రై చేసి
మేడం మీట్ చేయరా సౌండ్ వస్తుంది చేసారా
ఇంతే కనిపిస్తుంది.
ఎంత చేయండి ఆమె ఇచ్చాను సార్. సో దట్
సైన్ అవుట్ అని కనిపిస్తుంది. దాన్ని సైన్ ఇన్ చేద్దాం.
అదే
Very good morning, Varun. I'm Akila and I'm Panis. We welcome you all to the International Symposium on Bioinformatics training. And this is Therapeutics. Oh, this is Therapeutics.
స్క్రీన్ షేర్ చేస్తున్నారా?
నేను పెట్టాను.
Okay, ready for
Keep that microphone close to your mouth so that otherwise you are getting some, you are not able to hear.
So it's actually so basically not extended.
So your voice is breaking up.
Oh,
It is being disrupted.
they might yes. Sir, మీరందరూ కొంచెం మ్యూట్ చేసుకోండి సార్.
Dear all, our beloved respected register sir has joined. Please mute yourself. Whenever required, please switch on. Turn to unmute.
Good morning, sir. Very good
morning.
morning, Professor Kripanidhi Garu.
Good morning,
Professor
sir.
Kavi Kishore Garu.
Good morning.
Namaste.
Namaste. How are you, sir?
I am fine. I am 10,200 miles away from you.
True.
I am right now in the biotech hub of the world,
Oh, okay, sir.
Boston.
Oh, Boston, okay. Yeah, it must be pretty cold now there.
It is snow. It's about -8 degrees now.
Okay.
Pretty cold. Last couple of days it was pretty cold.
Right.
And I was very happy when I came to know there are 430 biotech companies in Boston.
Right, sir. Right.
Amazing. Amazing. I believe this is the hub. Once upon a time, California was the hub, I believe.
Right.
Now, Boston and Cambridge has become the hub.
Yeah. And Hyderabad is also, we have around 220 biotech companies, sir.
Yeah. Hyderabad is the pharma capital of the world.
Right.
Boston also, you are thinking about our biotechnology, sir. Okay, sir.
So what time we are starting? 9:30?
9, sir. 9.
9 o'clock. Okay.
Chairman sir is away. He's flying from Hyderabad to Guntur.
Oh.
So he will be joining us late by 9:40 or so.
Oh, okay.
But meanwhile, we can start he said, he would join. So
Okay.
yeah, he has asked us to start sharp by 9 o'clock, sir.
Yeah, no issue, sir. No issue.
Yeah. The flight lands by 8:45 at Vijayawada. So it may take an hour or so. So 9:40, 9:45, he should be on the campus.
Yeah. Okay. I will unmute.
Certainly, sir.
ఆయనతో ప్లాన్ చేసుకోండి మీరు ఆయనతో ప్లాన్ చేసుకోండి
So are you ready, madam?
Start. Yeah.
Shall I?
Very good morning, ladies and gentlemen. A very good morning, ladies and gentlemen. We will be commencing the event in a few more minutes.
నేను నొక్కటం ఏంటి హలో అబ్బాయిని పిలవన్నాడు అంత కోపం అయితే ఎట్లా బాబీ నన్ను
నమస్తే అండి ప్రొఫెసర్ నాయుడు గారు.
నమస్కారం అండి. నమస్తే.
In a minute, we'll be starting the program in a minute, sir.
Are you able to hear my voice?
Yeah.
Yes, sir.
Yeah.
Yes, sir.
We
Okay,
hear you. No problem. Absolutely no problem, sir.
Thank you.
It is very clear. We are also able to see you. No problem, sir. Absolutely.
Thank you. Nice of you.
Yeah. Chancellor will be Vice Chancellor will be joining in a minute, sir. So we are just waiting, and Chairman sir is on the way. So he will be joining by 9:40 or so.
Whether inauguration by him or by me?
No, Chairman will inaugurate, sir. So you will be releasing the book of abstracts, and you will be giving the message, sir.
But I got a message that the inaugural will be mine.
Chairman, in case Chairman does not turn up, then you can give your inaugural address, sir. No problem. But he said he is on the way. He would join us late by 9:40 or so. By 9:40 or so. So.
Now, my session to maintain the timing.
Certainly, sir. Certainly. We are
And
starting.
then let him give a message later on.
Okay, sir. Okay.
Because all the other people, scientists from abroad are also waiting.
True, sir. True. We'll start, sir. We are about to start, sir.
Yeah, okay.
So good morning, Chancellor sir, Krupanidhi sir.
Good morning, Professor Krupanidhi.
Warner sir.
Warner. Happy to see you.
Yeah, happy to see you also.
Thank you.
Good morning, Chancellor sir. Dr.
Good morning.
Raghunathan here.
Yeah, very nice. You're talking from USA.
Yes, sir.
So it's over. The purpose for which
I can hear you crystal clear. From Hyderabad to Guntur, for the board meeting, I can't hear you. But now I am 10,200 miles. I can hear you crystal clear.
You are also very clear. Your face is very clear.
Sir.
Yeah, so thank you. Nice of you.
Welcome, sir.
The program is over for which
yes, sir. It's over, sir.
So convey my congratulations and the blessings to the newly wedded couple.
Thank you, sir. Thank you so much, sir. Thank you so much.
Nice of you.
Good morning to all of you. Prasad here.
Good morning, sir.
Sir, good morning, sir.
Good morning. Professor Raghunathan Naidu, good morning. Prasad here.
Good morning, dear Vice Chancellor. Yeah, good morning.
A very good morning to one and all. I am Akhila. And I am Padmini. We welcome you all to the International Symposium on Bioinformatics for Disease Therapeutics, organized by Department of Biotechnology, Vignan's Foundation for Science, Technology and Research, the abode of great learning under the aegis of AP Bionet. It gives me an immense pleasure to welcome all the dignitaries, eminent scientists, technical giants across the world to this international information interchange to share their valuable experiences and discoveries with us. Ladies and gentlemen, it's a high time to have such an edifying event taking place to highlight the remarkable contributions of bioinformatics in the therapeutics for various deadly diseases. I'm extremely grateful to all the subject matter experts, area specialists joined over here to lead this technical banquet to shed some light on the gray areas which need potential focus. I'm sure you all will have fruitful and rewarding exchanges in the coming sessions. I wish you every success with this enlightening symposium and would like to take this forward with the prayer song. I would like to ask Ms. Vaishnavi for a prayer song. Ms. Vaishnavi, please.
Sa pa 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 ma pa ni ma pa ni sa mari sa sa ni pa pa ma ma ri sa ni pa ma ri sa ni ri sa Hamsavahini mrudu hrudayini manoranjani Vagdevi ve Hamsavahini mrudu hrudayini manoranjani Vagdevi ve Varala palukula nosagedu talli Veena pani namostute నమోస్తుతే వరాల పలుకుల నొసగెడు తల్లి వీణాపాని నమోస్తుతే సరసిజ నేత్రి శారద గాత్రి సృజన జనావణి వర ప్రదాయిని సరసిజ నేత్రి శారద గాత్రి సృజన జనావణి వర ప్రదాయిని కావవే వాణి చతుర్ముఖురాణి కీరవాణి నమోస్తుతే హంసవాహిని మృదు హృదయిని మనోరంజని వాగ్దేవివే వరాల పలుకుల నొసగెడు తల్లి వీణాపాని నమోస్తుతే వీణాపాని నమోస్తుతే సాస పాప పామ పని మప పాప ని సా మరి మరి సా సాస సని ని పప పమ మరి సా సాని ప మరి సా నిరి సా.
Thank you, Ms. Vaishnavi. Now, we shall have Jyoti Prajwalan by our Honorable Chairman, Dr. Lau Rataya Garu. Sir, please.
Good morning, sir.
Thank you. After lighting the lamp of knowledge, I would like to hand over the session to Dr. A. Rangarao, Organizing Secretary of the Symposium, Vignan's Foundation for Science, Technology and Research, to start his words by giving the genesis of InSyB 2020. Rangarao, sir, please.
thanks.
Your audio is bad, please.
So, hello, good morning to everyone. So, myself, I am Dr. Rangarao. So, working as associate professor in the Department of Biotechnology. So, it's organizing secretary for InSyB 2020. So, I welcome you, all of you. So, advances in life sciences have led an era of looking at biological problems through molecular level, and with further advances in biotechnology, we are now in a position to manipulate living organisms for a variety of social, industrial, agriculture, and medical applications. Modern day biology has turned more efficient in handling complex biology issues, including cancer, diabetes, crop improvement, and environment at a genomics level, thereby requiring high-throughput experiments which further generate high-throughput data. We need efficient approaches to store, manage, retrieve, and analyze this amount of data to answer many interesting questions, sensing the importance of computational biology, International Society of Computational Biology, and Asia Pacific Bionet education programs, annual conferences, and public talks. The AP Bionet has and able to make a significant impact by organizing such events, especially the International Conference of Bioinformatics. It was the 19th annual conference that was successfully held virtually last month. We from Department of Biotechnology and Bioinformatics, VFSTR University, have been very active in making researchers aware of various concepts of bioinformatics and helping them to increase their skills as well as impact their research. We have conducted several conferences, workshops, seminars since the establishment of this department, including the recent event, International Conference on Environmental and Sustainability, Human Health Development in December 2019, and also conducted a webinar on changing origins in combat COVID-19 in June 2020. We believe that InSyB 2020 will help in bringing many researchers working on various aspects of bioinformatics and the academic community on a common platform in order to drive the development of bioinformatics in the Asia Pacific region. InSyB, a sister event of bigger conference on bioinformatics, and INCOPE is a second annual conference series of AP Bionet with the aim of focusing on the thematic decisions targeting young researchers at a local community. Hence, with the expectations centered on the topics relevant to the local organizer, this initiative will not only create awareness and bring motivation among participants, but also develop a great platform for networking, collaborating, and scientific disclosure for academicians and industries from both international and, more importantly, local arena. This year will be the fourth iteration of InSyB with its inauguration in Jakarta, Indonesia in 2017, followed by Selangor, Malaysia in 2018, Punjab in India 2019, and this year again in India, but focusing on the south part, Andhra Pradesh. Despite the challenges of COVID-19, InSyB 2020 successfully attracted over 900 participants. The today event comprises a mix of keynote speakers, short talks, discussions, and meet the expert as opportunities, which will foster an interactive environment for integrated bioinformatics research and education. On behalf of the organizing committee, thank you very much for giving a wonderful opportunity. Thank you all.
Thank you, sir. That was very insightful. I'm very much delighted to invite Professor K. Ramurthy Naidu, Chancellor, VFSTR, former member of University Grants Commission, to announce the release of Book of Abstracts and to speak few words. Sir, please.
Akla, please take over.
All right, sir. Thank you. Now, I would like to request Professor K. Ramurthy Naidu to speak some few words. Sir, please. Sir, please unmute. Sir, could you please unmute yourself?
Hello? Is it audible?
Yes, sir, you're audible.
Yeah. Let me have the pleasure of releasing the Book of Abstracts of two-day International Symposium on Bioinformatics for Disease Therapeutics. So it is a good opportunity for the young budding scientists to know about this information, because any the seminar or a symposium, it would have the facility to exchange of the words, exchange of the ideas, exchange of innovative programs. So it is a good challenge for all the people. But still, I remember the bioinformatics and the biotechnology have emerged as emerging areas 3 decades ago when I was a Secretary State Council for Higher Education. Several private institutions have come forward to start with the bioinformatics and biotechnology. But I'm sure the Osman University started bioinformatics nearly 3 decades ago. Professor Kavi Kishore and taught for the students of bioinformatics in the MSc course. So I have a special attachment to the Department of Biotechnology. One, I taught MTech biotechnology students at Jawaharlal Nehru Technological University, 91-92, and also Department of Biotechnology at Vijnan University, one of the star departments, and has been organizing international seminars at least once in 2 years under the under the direction of Professor Krupanidhi, head of the department. I'm also happy to inform Professor Kavi Kishore, a distinguished scientist from the Osman University, has joined as the advisor at the Department of Biotechnology. So it is my pleasure to be associated with this symposium. I wish for the success of this international symposium for 2 days, today and tomorrow, and again wish you with the best wishes from me. Thank you. This is Professor Naidu. Thank you.
Thank you, sir, for such an enlightening speech. I would now like to request the Padma Shri awardee, the Vice Chancellor, Vignan's Foundation for Science, Technology and Research, the former Director, Space Center, Dr. M.Y. Prasad, to speak a few words. Sir, please.
Thank you. Hope I am audible. It's good morning to all of you. Respected Chairman of Vijnan Group of Institutions, Professor Ramurthy Naidu, Chancellor, Dr. Raghunathan, Registrar of the Vijnan University, Dr. Rangarao, Organizing Secretary of the conference, Professor Krupanidhi, Head of the Department, and all the distinguished speakers and keynote address participants from abroad. It's my great pleasure on behalf of the university to welcome all of you. And this International Symposium on Bioinformatics is being organized by the Department of Biotechnology in the VFSTR, which is deemed to be university, under the guidance of Dr. Professor Kavi Kishore, a distinguished professor in the field. Bioinformatics is an interdisciplinary field which has got the combination of biotechnology as well as the computer subjects. In fact, with such a fast growth of the computer field, the bioinformatics, what is taught a few decades back and what is being taught today has got entirely different character, because the artificial intelligence and machine learning have become the prominent areas in the computer sciences, which will be very useful in handling the data, especially large data, which is involved in the DNA sequence analysis. So the large data and the data structures, data streams, and the artificial intelligence together make a lot of difference to your work, which makes much faster your work in both analysis as well as in the processing of the data. Especially when it comes to the discovery of the new drugs and its rollout to the market, you need a lot of trials, both the theoretical simulations, then practical experiments, then clinical trials, which will prove whether your drugs are effective as you wanted or not. I think in this whole process, your added knowledge and added subject of computer science into your biotechnology will give you a lot of advantage. In fact, our country has got a large diversity. It has got a large poor people who need affordable health diagnosis and treatment. And we also have others who can afford healthcare on par with the developed countries in the Western world. You have to serve both sections. And more important, you have to definitely serve the people who need affordable cost. Healthcare at affordable cost, diagnostics at affordable cost. And as our Chancellor has said, the biotechnology department, the HOD, the faculty, and the students are very bright. As you can see from our presenters sitting there, and also the girl who sang the song excellently. And you are all the bright future of our university. I'm also very happy that the biotechnology department is organizing this seminar in the year 2020, which has got a special thing because the Nobel Prize in Chemistry is given this year to two lady researchers jointly. The first thing is, of course, they are researchers. I have nothing, I have no bias, but sometimes in the media they report some of the researchers in Western countries show some tendencies of bias against lady researchers. But this is excellent because Emmanuel Charpentier and Jennifer A. Doudna, both from Germany and USA, are jointly given this Nobel Prize for the genetic scissors. They have discovered genetic scissors, a tool to rewriting the code of life. In fact, they discovered CRISPR-Cas9 genetic scissors using the their research on the changes of DNA in the animals, plants, and the microorganisms with extreme precision. This allows you to do your personalized research on the diagnostics, customized research and diagnostics, and the discovery of the medicines very easy and very fast. And another very specific thing of this Nobel Prize is, I have not seen many people who got Nobel Prize in less than 70 or 75 years of age. But these two are people are relatively young. One is Jennifer is 56 and Emmanuel Charpentier is 52 years. So it was an excellent thing which had happened this year in the area of chemistry. And of course, in the area of medicine, Nobel Prize for Medicine also this year is very, very special because it is given for identifying hepatitis C, which is transmitted through the blood and affects mainly liver cancer. I think these two achievements are a recognition of the biotechnology people and pharmaceutical people and researchers who are doing biotechnology, bioinformatics, pharmacy, and the diagnostic research. This is the real celebration of your work. And in this year, when you are organizing an international conference with the participation of a large number of experts from different countries, is a great opportunity for our faculty as well as students to learn a lot from all of them and get your doubts clarified and build up a network where after maybe another 20, 30 years, we'll see somebody from you as well as good as them and recognized globally. With these few words, I thank Dr. Rangarao and Dr. Krupanidhi for requesting me to give this small brief address. And I wish all the best for this conference and very, very active discussions during your conference. Thank you very much.
Thank you, sir, for such a wonderful speech. I would now like to request Registrar, Vignan's Foundation for Science, Technology and Research, Commodore Dr. Raghunathan, to speak a few words. Sir, please.
Very good morning. Can you hear me?
Yes, sir.
Honorable Chairman of Vignan's Group, Dr. Lavu Rataya Garu, Honorable Chancellor of VFSTR, Professor Ramamurthy Naidu Garu, Honorable Vice Chancellor, Padma Shri, Dr. M.S. Prasad Garu. Good morning, sir.
Good morning.
All the respected guests for both India and abroad for this international symposium, participants, the entire team of Biotechnology Department of the Department of Biotechnology, BFSTR, a very good morning and hearty welcome to each one of you. At the outset, I should thank the organizers for having me for the inaugural session, even though I am very, very far from the university. I am sitting almost 10,200 miles away in a city called Boston, which is the cradle of biotechnology in the United States. There are about 430 biotechnology companies in and around Boston. Boston and Cambridge is just like Hyderabad and Secunderabad, is the twin cities. Within these two, Boston and Cambridge, there are about 430 companies. It is so coincident that I'm sitting here and I was asked to give a small 5 minutes session about the VFSTR. I am really thankful to the organizers. As regards the VFSTR is concerned, this is basically for those participants who are taking part in the symposium. It's a very, very young institution, a decade old, and founded by a very legendary institutional builder, Dr. Lavu Rataya Garu, way back in 2008. We started the university in the year 2009 as a deemed to be university. And within this short span of a decade, almost 10 years plus, the university has grown substantially with professional values, ethics, and honest growth. I think with a humble beginning of 230 odd students as intake way back in 2009, today I am very proud, ladies and gentlemen, to say that our intake has gone up to almost 10 times, 2,500 plus as a sanctioned intake, which covers all programs starting from science, engineering, technology, management, pharmacy, and very, very soon, we'll be having the law program also in the campus. That's the way the university has grown in these last 10 years. Now, as regards accreditation, we have two national agencies. One is the National Assessment and Accreditation Council and the National Board of Accreditation. Both these accrediting agencies have given their accreditation to the university. We have been both NAC as well as NBA accredited. And the National Institutional Ranking Framework of the Government of India has ranked this university in the top 100 in the country. I think this is no mean achievement considering the young infancy stage in which the university is presently situated. In addition, the Council of Scientific and Industrial Research of the Government of India has designated this BFSTR as a scientific and industrial research organization. Mind you, that gives a big credit for carrying out your research in frontier areas, including biotechnology. I think today the university is marching ahead with about approximately 480 faculty and equal number of non-teaching staff with about a student base of about 5,500 students in the campus. I think that speaks volumes about the way the university has taken shape, and much more has to be done, for which a very clear roadmap has been given for the founder and the management headed by the Vice Chancellor. And this roadmap has been converted as a strategic doctrine for the university for the next 10 to 15 years. I think this university has come a long way in these 10 years, and by providing quality education to all sections of the society, thereby lifting the socio-economic conditions of thousands of families in and around Guntur, Vijayawada, and other areas. I think this two-day symposium, which is organized by the Department of Biotechnology, as our Chancellor said right in the beginning, that this department has been organizing such symposiums, international symposium, once in 2 years, and this year they are doing it virtually. And I definitely, it's a very great platform. As our Vice Chancellor was telling, this is the ideal platform for the participants to discuss, debate, and mull over various issues pertaining to biotechnology and come up with some sort of a blueprint, which could be some sort of a roadmap for the department to proceed in the next 5 to 10 years. I think with these few words, I wish the symposium a grand success. And thank you very much once again for having me for the inaugural session. Thank you. Jai Hind.
Thank you very much, sir. That was very insightful. Now let me take pleasure to invite Professor S. Krupanidhi, Head, Department of Biotechnology, Vignan's Foundation for Science, Technology and Research, to speak a few words. Sir, please. Sir, could you please unmute yourself, sir?
Now, am I audible? Now, am
Yes,
I
sir.
audible?
Yes, sir,
Yeah,
you
yeah.
are.
So good morning to you all. So on behalf of the Department of Biotechnology,
Your link is frozen, please. You have a link disconnect. Thank you.
am I audible now? Yeah. So respected
Yes, sir.
chairman,
It is audible, sir.
yeah, respected chairman, Vignan Group of Institutions, Dr. Lau Rattay Garu. For those of the participants who are not familiar about Vignan Group of Institutions, I shall let you know at a glance. So Vignan Group comprises of nearly 41 institutions across the state of Andhra Pradesh and Telangana in India. So these 41 institutions are hosting students whose number ranges from 1,500 to 6,000. Direct benevolence of our chairman, Dr. Lau Rattay Garu. This is in nutshell. And beloved Vice Chairman, Dr. Lau Devraya Garu, and he is member of Indian Parliament, and distinguished Chancellor, Professor Kilari Ramamurthy Naidu Garu, and Honorable Vice Chancellor, Padma Shri Dr. M.Y. and our well-wisher, research advisor of our Department of Biotechnology, Professor Kavi Kishore. And again, our colleagues, friends, and beloved students. Good morning to you all. So for the time being, to begin with, I dwell upon the title of our conference, International Symposium, that is a few keywords which are there in our International Symposium are bioinformatics, disease, and therapeutics. So for the people who are not having in-depth knowledge in bioinformatics, it looks to be enigmatic how the bioinformatics will make us to understand the disease, will make us to understand the treatment modalities. That's what called therapeutics, for which I will give you a simple example. Somewhere in the month of November 2019, this coronavirus erupted. That time we did not name it as COVID-19. And somewhere in the month of March or February, almost we did not felt what is that called the pandemic. Later from March, after March, we felt the pandemicity. And up to that now, the technology was so refined and sophisticated that 20 nucleotide bases of positive RNA of the coronavirus was unfolded, unraveled. Not only the 29,000 nucleotide sequence of RNA of this coronavirus is unfolded, open reading frames are unfolded. Apart from that, the ribbon structure of the proteins of this coronavirus protein, spike, particularly the spike protein, was unfolded. So with this, we are able to identify through biochemistry, through pharmacology, through the chemistry, and through the microscopic examination. It only happened through docking, through this molecular docking studies. Okay, so like this, it is not enigmatic, it is true. So that bioinformatics is now having a magnificent edifice of software tools. As our Vice Chancellor sir is telling, there is a continuous progress of the computational field. Accordingly, bioinformatics is profusely. Sir was also mentioning about the Nobel Prize of 2020, about Jennifer and also the Emmanuel Charpentier, who did work with this CRISPR. CRISPR-Cas9 work also goes with bioinformatic tools, wherein this is a genome editing tool. In this genome editing tool, single gene disorders like thalassemia, alpha thalassemia, beta thalassemia, sickle cell anemia, and again Huntington's chorea, they all can be cured with this genomic, that is CRISPR-Cas9 genomic tool. That's what now, this is the year for, again, rejuvenation of biotechnology industries in 2020, because nearly 5 Nobel laureates are working on this. Okay, so after having gone through some aspect of the title of our international symposium, now it is my turn to extend an invitation to all our speakers. So there are nearly 16 talks that have been conceived in this program. Of these 16 talks, the major are COVID-19 talks. Nearly 30% of the talks are focused on this. The rest of the talks are on computer-aided immunotherapy, that is infection and immunity. The next one is on this immunoinformatics. In addition to that, there is a structural bioinformatics. Is it relevant? Is it relevant for this today? Yes, very much relevant. So that's what this receptor binding domain, the spike protein anchoring to ACE2 receptors was done only through the structural bioinformatics. That's what the importance of the bioinformatics is going to be highlighted, elucidated in this conference. I request all the participants to be attentive. Why? Because we forgot Spanish flu, which came in 1918. We also forgot smallpox. Now there's no, it totally eradicated by WHO. Now, in another 5 or 6 months, we are totally forgetting this COVID-19 because vaccine is coming and herd immunity will get developed. Nobody bothers about the COVID-19. So before the people forgot about the COVID-19, by attending these conference proceedings and deliberations, please find the lacunae in COVID-19 and see that you can contribute something so that your contribution will remain in this COVID-19 literature. Okay, so there's another important point I wish to let you know that in the Department of Biotechnology at Vignan University, for the past 5 to 6 years, we have been doing immense bioinformatics work, and 3 PhD theses are out. in this bioinformatics, particularly the molecular phylogeny. One of the speakers is doing on the molecular phylogeny of pulmonates, and he has done both nuclear genes and again the mitochondrial genes markers and elucidated the taxonomic relations and phylogenetic affinities among the pulmonates. Okay, in addition to this, I wish to let you all know that our Department of Biotechnology also has contributed nearly 10 publications in COVID-19, and as a result, our papers are all cited well. You all participants, I wish you all to see our publications in the net. And I wish to let you know a few points relating to the COVID-19. Our registrar sir is there in Boston now, Boston, California. Those areas are of low, what is it called, the temperature? Not only the low temperature, they're also having low sunlight. As a result of the low sunlight, vitamin D, they will be facing deficiency. So that's what one of the treatment modalities, that's what therapeutics, that goes with our title, is that the vitamin D deficiency encourages COVID-19 prognosticity. So that's what our treatment, our doctors are giving vitamin D tablets to those who are suffering, and those who are there in quarantine are made to move under sun for some time. So why? Because vitamin D is very much essential in this COVID-19, fighting against the COVID-19. Why? Because so there are vitamin D receptors, VDRs, vitamin D receptors on all WBCs, white blood corpuscles, which are myeloid and lymphoid progeny cells. So these VDRs will promote TLRs, toll-like receptors, to fight against the COVID-19. That's what innate immune potential will get reduced with the vitamin D deficiency. So that's what good amount of the information is passed through in this line. I wish the participants to take up this type of the work relating to vitamin D and viral infections. So with these few words on the topic of our international conference, now I'll let you know some aspects of the Department of Biotechnology. So our Department of Biotechnology came into work in the year 2006. Since then, we have been continuing, and we are having nearly 30 faculty members in the Department of Biotechnology. We are funded by Department of Science and Technology, Department of Biotechnology, and again, Ministry of Environment and Forest, that is from the central government. And we're also having nearly 600 students in the 3 programs that we are running. That is 4-year program of BTech Biotechnology, 4-year program of BTech Bioinformatics, and 2-year program of MTech Biotechnology, and again the research programs. Okay, so in this, we are having an unique feature exclusively in Vignans University that's called offering. We are providing a sort of an option to the students, that is internship. As a result, our students go to the industries, and also the faculty are also introduced to go to the industries to get acquainted. As a result, none of our faculty are obsolete. They're up to date with the industrial applications. That's what, wherever you go in Hyderabad, wherever you go in Bangalore, our Vignan students are represented. Biotechnology students are represented. So that's what I wish to let you all know. This is one of the aspects that's going on in the Department of Biotechnology. In addition to that, our Department of Biotechnology is having sophisticated equipments for biochemistry practicals, for biochemistry related experiments, for microbiology related experiments, and again, the molecular biology related experiments, particularly with regard to gene annotation and all those things. And in addition to that, we are having clean room facility in the Department of Biotechnology, which is having a facility wherein 5 micron sized particles are not existing there. Only 3 micron sized particles are present. As a result, it is so pure, a pure vicinity with which all animal cell culture experiments can be conducted here. In addition to that, we're also having the animal house approved by CPCSEA, and also we are having the biogas plant wherein 1000 kg food waste is being fed to the biogas plant. As a result, we are getting 16 kg into 6 gas cylinders every day. As a result, it is being used continuously in our hostel kitchens. In addition to that, the faculty of the Department of Biotechnology are maintaining a medicinal plant garden. So wherein more than 20 species of medicinal plants like Mucuna pruriens and several other plants, Andrographis paniculata, stevia, they're all existing in our medicinal plant garden. With all these things, I wish the participants will be having very nice time in this 2-day long international symposium on bioinformatics for disease therapeutics. And in this, I wish you all participants to visit physically our Department of Biotechnology, Vignans University, at your convenience. We're happy to have collaborations. We're happy to have, what is it called, interactions with you all, either both academics and also in the research. Particularly with regard to the academics, what I wish to let you know is our syllabus is getting revised once in 3 years. And our syllabus is latest and also contemporary to the industrial developments. That's what I wish to let you know here. Immunoinformatics, immunotechnology, phase display, and again, vaccinology. These are all the 4 courses we introduced to our students in R19 regulation. As a result, our syllabus is on par with the industrial developments. So that's what we always wish to have collaboration, those who are interested for academic and research activities. So I wish you all have a nice time, enjoy the conference in the deliberations and also in regular proceedings. Thank you one and all.
Professor Kripanidhi Prasad here.
So thank you.
I see a lot of distinguished participants like Dr. Narhari Sastry and Dr. Kenta Nakai.
Yes,
So
sir.
can you or Rangarao can briefly mention about their expertise so that all the participants will know and then they will be attending all these sessions with them. Thank you.
Sir, Vice Chancellor, sir, when shall I do, sir? Now itself?
Dear sir,
All people will know, okay?
After VC, sir, technical session is there, sir, wherein our anchors will introduce the invited and keynote speakers, sir.
No, you can mention all of them and their specialization briefly in this session. That is appropriate because all the people will know. Okay. Thank you.
Thank you, Vice Chancellor, sir. Dr. Anil, please provide me the chance. I will, as per the advice of our Vice Chancellor, sir, I will try to introduce our invited speakers and the keynote speakers. Shall I proceed, Anil Kumar? Anil Kumar. Anil Kumar. Rangarao, sir.
Sir, good morning, sir. Sir,
Yeah.
we will give you within 2 minutes, 2-3 minutes, we will give you the details.
To whom, how do you give me, ma?
We will give you, sir, within 2 minutes.
No, do you give me the write-up, ma, or what, ma?
Professor Kripanidhi,
sir.
you can mention their name, their
Yeah.
organization, and their specialization.
Yes,
That will be sufficient at this level. Okay.
Very good, sir. So, again, dear participants of our international symposium on bioinformatics in relation to disease therapeutics, we are glad to have
Dear participants and gentlemen, we really apologize for this glitch in internet. Please bear with us for a few minutes. Thank you.
Some problems.
Hello, good morning to all of you. So myself, I am Dr. Rangarao. So Dr. Kenta Nakai is from Japan, and he is working in Kyoto State Kyoto University, National Institute of Basic Biology. So Professor Dr. Kenta working on human genomics related research, and mainly is working on computational ways interpreting biological information, especially that of transcriptional regulation from genome sequence data. Actually, he's an expert in genome sequence data, sir, and human genomics. And whereas Professor Nahim Shastri is working on mainly focusing on computational chemistry, computational biology, and computer-aided molecular design, chemoinformatics, and big data, and artificial intelligence related areas. Professor G. Narasimha Shastri is working on specific areas, sir.
What more? Some more people you can say? Hello?
Yes, sir. Yes, sir.
You can tell the other foreign, the distinguished scientist.
Yeah, yes, sir. And Professor Dr. Bruno Guetta is focused on bioinformatics engineering aspects. So he's from Australia, the University of New South Wales, Australia.
Okay.
And then Professor Ramaswamy is from Singapore. So he's working on, he's a senior research scientist, Genome Institute of Singapore. So he's focusing on specialized statistical genetics, bioinformatics, and code monkey and p-value generator. So especially he's working on that aspect, sir. Professor Ramaswamy.
Rangarao sir, please allow
Yes,
me
sir.
to speak. Please allow me to speak, sir.
Yeah, please, sir. Please.
Yeah. So, as we indicated, you all participants, that there is a great momentum moving around now about COVID-19. There are nearly more than 5,000 articles have been published in the short span of 8 months on COVID-19. None of the other topics in biotechnology has taken so much attention. And as I indicated to you earlier, in a matter of another 5 to 6 months, everybody shift their field from COVID-19 to the other viruses. That's what I request all participants to go very carefully the deliberations that are going to be deliberated by our experts. In this line, we are happy to have with us the keynote speaker, Dr. Kenta. He has worked with Kyoto University, wherein in Japan, last 2 years back, we had received a Nobel Prize from Kyoto University, that is Honjo Tasuku, with regard to the, what is it called, blockade of immune checkpoint therapy. So he has done good amount of work on NGS tools. NGS tools are the order of the day now. So wherever you go, the multiplex PCRs and the amplification and the disease diagnosis is being done with the NGS tools. And we're also happy to have amidst us Narahari Sastri Garu. Narahari Sastri hails from he is a Bhatnagar awardee. Bhatnagar awardee, one of the reputed award that our Indian scientists secure from the Prime Minister of India. And he has been working on infection and immunity as indicated in my talk. And he is the director, CSIR Northeast Institute of Science and Technology, India. Infection invariably prompts immunity, because COVID-19 is prompting us to raise the herd immunity and also the individual immunity. It involves several of the innate and adaptive immune processes in which the immunoinformatic tools and adaptive immune processes, everything has been elucidated by our Dr. Narahari Sastri. We are delighted to listen Dr. Narahari Sastri Garu. And we're also having yet another speaker, Dr. Bruno Greta. So Dr. Bruno Greta is a bioinformatics engineer. So why we are speaking of the bioinformatics engineer means here we are constructing the ribbon structures. That's what the structural bioinformatics, wherein the structure earlier in our India, we used to have G. N. Ramchandran in Chennai, who has done a marvelous work relating to the structural bioinformatics. When there was no computers, he has done in Chennai Madras University. So that's what called Ramchandran plot. Even now, all over the world, globe, it has been Rama plot is being taught, is being used. So like that, the structural bioinformatics, particularly the protein expression, all those things are being dealt by Dr. Bruno Greta. We're also glad to have with us Ramaswamy, Professor Ramaswamy. Professor Ramaswamy is from the University of Oxford, and he has done good amount of work on genomes of viruses and other fields in the area of viruses and again the plants and animals. So we are glad to have this many bioinformatic tools exposed to our participants. All the participants, I request you all to go through. In addition to that, from our Department of Biotechnology, we are having excellent speakers on this particular theme. That is, Dr. Vijay Sai is going to speak on molecular phylogeny of pulmonates. Dr. Anil Kumar is going to speak on COVID-19, and even our Abhinav Parashar is going to speak on COVID-19. So with this few words about introducing our revered experts for this international symposium, I now pass on the mic to my colleagues. Thank you, one and all.
Thank you.
Thank you very much, sir. That was very informative. Now let me invite convener of the symposium, Dr. N. Jalaja, Department of Biotechnology, Vignan's Foundation for Science, Technology and Research, to give her valuable words about this InSyB 2020 and AP Bionet. Ma'am, please.
Greetings to all. This is Jalaja, Associate Professor, Department of Biotechnology, Vignan Foundation for Science, Technology and Research, Dignitary University, Guntur, Andhra Pradesh, India. So welcome to International Symposium of Bioinformatics for Disease Therapeutics, being organized under the aegis of AP Bionet and the Department of Biotechnology, Vignan Foundation for Science, Technology and Research University. So, respected Honorable Chairman Sir, Vignan Group of Institutions, Dr. Laura Garu, Sri Krishna Devarayul Garu, Vice Chairman, revered Professor K.R. Naidu, Chancellor of Vignan's University, Padma Shri, Dr. M.Y.S. Prasad, Vice Chancellor, Vignan's University, Dr. M.S. Raghunathan, Registrar, Dr. A. Rangarao, the Organizing Secretary of this two-day International Symposium, Professor Krupanidhi, Head, Department of Biotechnology, respected Dr. Muhammad Asif Khan, President, AP Bionet, Professor P.B. Kavikishore, Program Coordinator, the keynote speakers and invited speakers of the symposium, faculty members, scholars, and students. It gives me great pleasure to stand before you as an organizing committee member and to welcome you all on behalf of the organizing committee to the Vignan's Foundation for Science, Technology and Research, Guntur, the place of higher learning. I'm glad to be a part of International Symposium on Bioinformatics for Disease Therapeutics being organized by the Department of Biotechnology in collaboration with the Asia Pacific Bioinformatic Network, that is AP Bionet. Thank you, Dr. Asif Khan, for disposing your faith and confidence on the faculty members of Department of Biotechnology and Bioinformatics, Vignan University, in organizing this event. International Symposium on Bioinformatics is a flagship symposium and an Asia-specific bioinformatics network. The current InSyB 2020 event is the fourth in the series and for the first time virtually, given the pandemic situation. So the last three InSyBs were held in Indonesia 2017, Malaysia 2018, and in Punjab, India 2019. Although this is the second time InSyB is being held in India, this time it is being held in the southern part of India. About AP Bionet and InSyB, founded in January 1998, AP Bionet is dedicated to the advancement of field of bioinformatics with an Asia-specific focus. AP Bionet has organizational and individual members from over 27 countries in the regions, from industries, academia, research, government, investors, and international organizations. Its mission since inception has been to pioneer the growth and development of bioinformatics, awareness, training, education, infrastructure, resources, and research among member countries and economies. It is spreading in a number of key bioinformatics initiatives in collaborations with our international organizations and industry partners, specifically improving the bioinformatics network infrastructure, facilitating the exchange of data and information, developing training programs, workshops, conferences, and symposia, and promoting regional collaboration in the field of bioinformatics. The InSyB bridges the gap between INCOPE, International Conference on Bioinformatics, and regular local AP Bionet supported workshops or the training courses. The two-day annual InSyB is expected to be centered on topics relevant to the local organizations. A mix of keynotes, short talks, discussions, poster sessions, and meet the expert opportunities shall foster an interactive environment for integrated bioinformatics research and education. Let me briefly tell you about the history of InSyB. The first inaugural InSyB was held in YRC University, Jakarta, Indonesia in 2017. So InSyB 2017, the Asia-specific Bioinformatics Network is a non-profit organization with a mission for advancement of bioinformatics in the region. This year, to commemorate the 50th anniversary of YRC University and also to promote bioinformatics in the region, YRC University, AP Bionet, and Perdana University are going to collaboratively organize the first international symposium of bioinformatics in 2017. So this symposium would be second collaboration between Perdana University and YRC University following the MOU signed on 8 January 2016 and the first with AP Bionet. This symposium aims to spread awareness and development of bioinformatics in Asia Pacific, especially Southeast Asia region, and also to strengthen the relationship between Perdana University, Center of Bioinformatics, Asia Pacific Bioinformatics Network, and YRC University. InSyB 2017 will serve as an intermediate milestone in preparing YRC University to bid and host the international conference on bioinformatics in INCOPE in the near future, to be held for the first time in Indonesia. InSyB 2017 takes theme of bioinformatics for precision medicine and will include session on higher performance computation and big data analysis. Then InSyB 2019, advances in life sciences have held on era of looking biological problems at molecular level, and with further advances in biotechnology, we are now in position to manipulate living organisms for a variety of societal, industrial, agricultural, and medical applications. The modern day biology has turned more efficient in handling complex biological issues, including cancer, diabetics, crop improvement, and environment at genomic level, thereby requiring high throughput experiments, which further generates a high throughput data. We need efficient approaches to store, manage, retrieve, and analyze this mammoth of data to answer many interesting questions. Sensing the importance of computational biology, the ISCB as well as AP Bionet has been playing a vital role in development of the scientific discipline through a number of ways. AP Bionet has been able to make significant impact by organizing many brand events, including InSyB. The prime aim of International Symposium InSyB 2020 is to bring scientists of international repute on a single platform to discuss about the current human healthcare system and to bring awareness about the importance of bioinformatics in therapeutic interventions. I'm certain that the theme of InSyB 2020 covers the whole spectrum of disease therapeutics, especially viral diseases like COVID-19, utilizing the tools and techniques of computer. I am hopeful that we will be meeting through this webinar many renowned scientists, pharmacists, academicians, and industrialists, and stress the need to accelerate the discoveries associated with human disease such as cancer, tuberculosis, and viral infections. COVID-19 pandemic has not only cautioned but also challenged us what a small vital particle can do the human existence itself, and certainly it is a challenge for all of us to deal with the viral particles than any other disease-causing organisms. Time is right for us to arise and awake and to combat with the microbial systems that are leading to such devastating pandemics. There is an immediate need to sense about the enormity of the problem and to utilize all our resources of computational tools and techniques for evolving solutions that help humanity from the brink of extinction. Taking the above situation into cognizance, InSyB meetings under the guidance of international scientists have been playing pivotal roles in expanding and disseminating the knowledge base by conducting seminars, organizing educational programs under the umbrella of AP Bionet. The Department of Biotechnology and Bioinformatics, VFSTR University, has been very active in the area of bioinformatics and biotechnology and helping the students of this rural area in making a career in this crucial area of computational biology. Realizing the importance of bioinformatics long ago, our chairman and vice chairman has introduced the courses in bioinformatics at graduate levels and also the PhD programs. I'm glad to inform you all that this symposium is witnessing 7 renowned international speakers besides 10 national speakers covering a range of topics but fit into the theme of symposium. I'm equally happy to place it on record that 964 participants have registered from across the Asia-Pacific countries. This only emphasizes that students are interested and enthusiastic to learn more about the computer tools and algorithms that are applied to derive meaningful information by deploying computer tools to biological databases and to DNA sequence databases. Stimulating discussions with experts in the field would certainly enrich our minds and help us to develop more international collaborations. The two-day International Symposium InSyB 2020, I am sure, would help the participants in many ways and help the students to take up the bioinformatics as a career. On behalf of the organizing committee, I'd like to thank all the speakers who have readily agreed to participate and disseminate their knowledge through this forum. I also like to thank all the registered participants for their interest and enthusiasm to understand how computer tools are being applied for therapeutic interventions. Thank you very much for giving me an opportunity. Thank you.
Thank you very much, ma'am. At this point, I would like to thank all the internal dignitaries for all that they have spoken. You have all heard about what the eminent speakers would be speaking today and who they are. For all those minds that are a little more inquisitive, I would like to invite Dr. Jagdish Kumar to introduce our first speaker, Dr. Kenta Nakai. Sir, please.
Thank you, Akila. Good morning to all speakers, guests, participants. Greetings of the day. Dr. Kenta Nakai was born in Osaka, Japan in 1963. He received the PhD degree on the prediction of subcellular localization sites of proteins from Kyoto University in 1992. From 1989, he has worked at several institutions, including Kyoto University, National Institute of Basic Biology, and Osaka University. From 1999 to 2003, he was an associate professor at the Human Genome Center, the Institute of Medical Science, the University of Tokyo, Japan. Since 2003, he has been a full professor at same institute. His main research interest is to develop computational ways for interpreting biological information, especially that of transcriptional regulation from genome sequence data. In 2020, he was awarded the first JSBI prize for his research activity on genome and protein sequence analysis, especially for prediction of subcellular localization signals by Japanese Society of Bioinformatics. Sir, Dr. Kenta Nagai, sir, the session is yours, sir.
Thank you very much. So can I share my slides?
Yes, sir, you can share your slides, sir.
I can't. You must change the setting of the Zoom, I think.
Sir, you can share now. You can share your PPT, sir.
Yeah. Now, yes. Can you see the slide?
Yeah, yes, sir. It is visible, sir.
Okay. So, and do you hear me?
Yeah, sir. It is audible,
okay.
Sir.
Yeah, thank you very much for kind introduction, Dr. Kumar. And so I'd like to also thank the organizers of the symposium for inviting me to this time. I'm very happy to hear. So let me start my talk. So as you know, the technology of DNA sequencing has made great progress in these 20 years. So then the cost for determining the human genome sequence has reduced very dramatically. According to this slide, the cost for determining a human genome is around $1,000 US, but according to the announcement of some developers, the cost will even become reduced to $100. So soon we will see the new age of $100 genome era. So that will inevitably change our society. That is, the new era of personalized medicine will come. In this personalized medicine approach, the genome sequence of every person, every individual is predetermined and stored. So, and the information of each individual's DNA sequence, genome sequence, the information will be used for the prevention, diagnosis, and treatment in daily medicine. So it's very important to see the difference of the genome sequences of individual. That kind of differences are called variants. Some variants are known to be associated with diseases. So that is the people who have a certain types of variants are known to be more susceptible to become a certain disease. So it's important to understand the meaning of each variant in the genome. As you know, the variants can occur in various places in the genome. Some variants are seen on the protein coding regions of the genome, while the other variants are seen on the non-coding regions in the genome. And generally speaking, it is more easier to assess the meaning, the importance of the variants occurring on the protein coding regions, because we don't know how the genetic information is encoded in the non-coding regions. Well, in the non-coding regions, some information about gene expression regulation or splicing regulation, RNA splicing regulation are seen. And but as you see in the slide, some disease-associated variants are seen frequently in the non-coding regions. So we must figure out how to assess the importance of the variants occurring on the non-coding regions. Because it's relatively difficult to assess the importance of the variants occurring on non-coding regions, people are using additional information, such as the epigenetic information. In this case, the post-translational modifications of histones, such as the H3K4 trimethyl modification in the histone. One of the purposes of the main purposes of this address is to point out that the next generation sequence techniques are not only used for just reading the genome sequence or DNA sequences, but also be used for various purposes, that kind of getting various kind of information, such as the histone modification information. Here, this is an example. The histone modification information, as well as the binding information of transcription factors on the genome, can be obtained by the technology called ChIP-seq, the chromatin immunoprecipitation. And so adding this kind of information, that many people have constructed a system to assess the relative importance of the variants occurring on the non-coding regions. And our group has also published a paper recently on such a system. We used a semi-supervised deep learning approach for predicting the functional effects of genomic non-coding variations. But we will not go into the details of this approach now because of the time limitation. And so, as I told you, the technique of NGS can be used in various purposes, for various purposes. Another technique for using the NGS is ATAC sequence. The ATAC sequence is used for detecting the DNA region, genome regions that are loosely packed or open, the so-called open chromatin. You know that chromatin, DNA, genome, human genome are packed with nucleosomes and make an chromatin structure within the nucleus. And the actively used regions in the genome take a more loosely packed conformation and called open chromatin. So, and such kind of open chromatin regions can be detected by techniques such as ATAC-seq. The ATAC-seq uses transposons, and transposon preferentially binds to the open chromatin region. So, using this nature, we can map the positions of in the genome where the open chromatin structure is seen. And so here is one example of our works based on the ATAC sequence. In this work, we reported that using the ATAC sequence, the sequencing, we can more efficiently detect the contaminated bacterial sequences from the host genome sequences. You can see in the graph, in the right, the ATAC-seq is more useful for detecting the contamination of bacteria. The reason of that is that the bacterial genome, the bacterial chromatin is more relaxed, the loose structure compared to the chromatin structure of the host genome. So if we use the ATAC-seq, the ATAC, the transposon, preferentially sequence attacks the bacterial genome. So the efficiency of detecting bacterial genome will be increasing. So that's why the ATAC-seq can be used to enhance the efficiency of detection, contamination detection. Anyway, we need to think about how to construct a system that can efficiently interpret the genome sequences, the variation, the meaning of variants on the genome. And here, the one of the difficulties, the reasons of the difficulties why the interpretation of individual genomes on the variants in the non-coding regions is that they have many exceptions. So the reason our current genome has not been designed at this current stage, but has been evolved from a very simple form to the current form. The situation is similar to the natural language, like English or Japanese. So you know that the Japanese or both Japanese and English not or Hindi are not designed in the current form. They have been evolved in the course of history. So the many words have disappeared or appeared during the evolution. So and the language, our grammar contains many exceptional cases. So not a very simple grammatical rule can explain everything. So this is very similar to our situation in the genome. So so when I was a graduate student, I think about how to construct a system that interprets the genome sequence. And I thought that maybe a technique in artificial intelligence called knowledge-based systems may be suited to as an interpretation of genome sequences that contain many exceptions, complications. So this is an example of a famous export system, the knowledge-based system called MySyn. The MySyn is a system that can diagnose the patients if he or she is infected by an bacterium or not. So, and the system contains many kinds of if-then type rules like this. And so combining the rules, the system diagnoses the patient and makes some comments, advices from their treatment. And I thought that this approach can be used to interpret the genome meaning of the genome sequence. At that time, I focused on the interpretation of the protein subcellular localization information on protein sequences. That is, that each newly synthesized protein in the cell are recognized with their protein sorting signals, such as signal peptides, and then be transported to the final subcellular localization site, such as the nucleus, mitochondria, inner membrane or outer membrane matrix, etc. And that kind of final localization site is determined by the combination of various sequence features of the sequence, the amino acid sequence. The rules can be written in this way if we use a natural language. So collecting this kind of knowledge in the system, we can interpret the newly input amino acid sequence and predict the final localization site. So about 30 years ago, I made such a kind of an export system that named it PSOLT. And the PSOLT system had about 100 if-then rules and could predict the final localization, subcellular localization of an input precursor amino acid sequence. And but the one problem of the PSOLT, the original PSOLT, was that the maintenance of the knowledge base was not easy. Because of the genome sequencing era, so many newly determined sequences increased. But the optimizing the system with the increase of the training data was not so easy. So, and the mainstream of the artificial intelligence also has shifted to the machine learning. So with the collaboration with Dr. Paul Horton, we used an technique, k-nearest neighbor method in machine learning to make the prediction of subcellular localization site. And this is an PSOLT2 or Wolf PSOLT. Both of the systems use the machine learning approach, so it's easy to be updated with the new training data. But the merits of explicitly including many exceptional knowledge have been lost. And so the PSOLT has been released through the internet, and many people have used it. The main 3 papers of the PSOLT system have been cited more than 1,000 times. So this was rather successful. So I wonder if a similar approach can be used for the interpretation of the non-coding regions, the transcriptional regulatory regions of the genome. As you know, the genome, all organisms start from a fertilized egg, and the fertilized egg divides and repeats the division, cell division, and that divided cells differentiate and make an complicated body structure consists of many types of tissues. So, but the important thing is that each cells in the tissues has the the same genome sequence. So that's still mysterious why the same genome sequence can indicate the making of so complicated different gene expression patterns. And here is one proposal that in a certain types of in the genes that are co-expressed, expressed in a similar way. Maybe, for example, some genes are exclusively expressed in a certain tissue and are not expressed in other tissues at all. But that kind of cell type specific or tissue specific expression of genes are often seen. And if we look at the regulatory regions of these genes, maybe we can find some common structure. Because these genes may be regulated by related, not the same, but maybe related mechanisms. So they may share, or the majority of the genes, or the some of the genes at least, may share the common structure in their regulatory regions. Here is an example that this is a schematic representation of the upstream regions of the muscle-specific genes. Each upstream regions has several cis elements. The cis elements are actually the binding sites of transcription factors, like MyoD, MEF2, etc. So, as you see, that not all upstream regions structures are the same at all, but they are more or less related. So if we use a set of rules, maybe we can distinguish this group of genes with the other genes. So with this idea, Alexis Vandenboom, who was a PhD student of my lab many years ago, constructed a system for the interpretation of upstream regions and for the prediction of tissue-specific expression of genes of various species, including mouse and human. And his system used a set of genes, set of rules, the small number of rules that describe the group of co-expressed genes. Here and the rules were automatically optimized, chosen by using the genetic algorithm. Here is an example of the rules that he found. This is an example for the rules that describe the features of the upstream regions of the genes that are specifically expressed in human fetal liver. So it's a human fetal liver model. And so the first rule shows that this motif is found near the TSS, transcriptional start sites. And another rule, the second rule, tells that this motif is found near another motif, this. So the two motifs, a pair of motifs is found within a certain threshold range. And the third one, similar, etc. So combining these kind of rules, we can distinguish the group of genes that are specifically expressed in fetal liver, looking at their upstream regions. And so the motifs that were first recognized by the so-called de novo motif finding algorithms, such as Gibbs sampler or etc. But actually the found motifs, some of the found motifs correspond to non-binding site motifs of the non-transcription factors. And we systematically applied the algorithm to the many tissue-specific genes, and about half or more number of the tissues we successfully made a rule-based system to detect the tissue-specific expression of the genes, input genes. For example, here we see that the genes that are expressed in the tongue or the fetal liver or the kidney, skeletal muscle liver, these genes were relatively easily recognized. The liver-specific genes were relatively easily recognized from others. However, not all tissue-specific genes were recognized by this approach. Maybe there are some reasons. But actually, the model constructed using a certain organism. So for example, here, the prediction model based on the human data, or if it is applied to the mouse sequences, the tongue-specific model, human model is, if applied to mouse sequences, the upstream sequences, that it could successfully, statistically meaningfully recognized tongue-specific genes in mouse also. So that shows the validity of our approach more or less, and also the promoter structure, the cis-regulatory structure tend to be conserved between species, the mammalian. But one weakness of the our approach that is that we couldn't consider the effect of distantly located cis-regulatory elements, such as the enhancers, known as enhancers. But currently, it is believed that the enhancers are more responsible for determining the cis-regulatory tissue or cell type specific expression of genes. So later, we have mainly focused on the analysis of the smaller organisms with smaller genome size. For example, Arabidopsis, fruit fly, and C. elegans. In these organisms, the size of the non-coding regions is relatively small. So it's unlikely that we miss the distantly located enhancers with our approach. So another, my former PhD student, Yosvani Lopez, has analyzed, published several papers. One example is here that he analyzed the antenna-specific genes in Drosophila, fruit fly, and found some common structure on their promoter regions, the upstream cis-regulatory regions. And he not only analyzed the tissue-specific expression genes, but also he analyzed the developmental stage-specific regulatory genes. Some genes are specifically expressed in a certain developmental stage. So that kind of information can be also used to assess how some of the genes, these co-expressed genes, share the common structure in the upstream regions or not. He successfully found some common structures and reported it in a paper. And more recently, new technology called single-cell RNA sequence has appeared. The technology is still under development, but with this technology, you can see the transcriptome, the expression pattern of genes in a single cell level. That is a great news for us, because we can even if we say that a gene is specifically expressed in a certain tissue, but certain tissue consists of various kinds of cell types. For example, in the muscle consists of various kinds of cells. So if we see the gene expression in a single cell, the cell type, I think the accuracy of the detecting or the precision of detecting the common structure will increase because of the accuracy of the data increases. But on the other hand, the RNA single cell RNA seq is still notorious their high noise. So there is an trade-off between them. But here we report our current attempt. Unfortunately, this is not finished. It's still ongoing project. So I can only show its preliminary result. But please have a look. The more recent, they are more recent attempts. Actually, one of my lab member staff, the AG Nagai, Dr. AG Nagai has analyzed the large-scale C. elegans single-cell transcriptome data. The original data was published last year in Science that contained 89,000 cells data. And here is an result of the cell clustering. And we not only produce the cell clustering like the original paper, that we also tried an clustering of genes by based on the cell type specific expression of the transcriptome data. In other words, that we transpose the matrix, the expression matrix between the genes and the cells. The genes and the cells, the rows and columns can be transposed in the by way of the genes and cells. So then we can get the cluster of genes that are expressed in a similar manner. So we mapped the obtained clusters on the clustered cells. So if we use an k-means clustering arbitrarily, the found cluster of genes with a similar expression pattern is mapped in a certain cell type like this in the upper slide. So it means that this cluster of genes shows a cell type specific expression of genes. So we analyzed. We analyzed the gene ontology terms of the each cluster, the co-expressed genes, and of course they are still a preliminary result. But fortunately, we could see that this slide is too small to read. But we observed that each cluster is characterized with a certain geo term. In this slide, the x-axis shows the relative statistical significance of the geo term enrichment, and the size of the circle means the number of the genes detected with having that term. So the different colors shows the different cell types, different gene cluster. So that means that the different cluster, each cluster of different cluster is characterized with different geo term. So they have different functional activity. And if we shuffle the clusters of the genes, of course we didn't see the enrichment of the geo terms like this. And also, okay, may I continue?
Yes, sir, you can continue, sir.
Okay. The motif enrichment, we also checked the motif enrichment. not only the geothermal enrichment, in the cluster of genes, we checked if the upstream regions of these clusters share the enrichment of a certain motif. And this time, we didn't use the other new motif finding algorithm, but just use an existing motif database like Jasper. But we found that some motifs are enriched in the upstream regions of certain cluster. Like this here is one example of muscle-related cluster, and here is another cluster, the cilium and sensory function motif cluster, gene cluster, and they share some characteristic motif on the upstream region. So it's still, we are still in the current stage, but if we believe that if we continue our attempts to characterize the upstream regions of these clusters, that we will finally make a very comprehensive map of the upstream regions that characterize the cis-regulatory element type architecture of the gene regulatory regions. And that will be useful for assessing the variance of the genome sequences. Okay. But it would be too optimistic if we believe that that kind of cis elements based simple approach will be suffice to understand all the cell type specific expression of genes. Actually, it is also believed that the cell type expression of genes is regulated by the changes of the higher order chromatin structure like this here, as shown here. In this example, a loop structure in the chromatin is changed between different cell types like this with the cohesion ring, the movement of the cohesion rings, and then the target genes of enhancers can be changed between different tissues, different cell types. So if this is true, but maybe it is true more or less. So we should include this kind of mechanism to understand the importance of the genome sequence. And also, it is observed that the genome sequence, the chromatin structure, chromatin is divided into two components, the A compartments and B compartments. The A compartments of the genome is located near the center of the nucleus and takes a relatively loosely packed configuration and open and actively used, while the B compartment of the genome is located near the periphery of the nucleus and tightly packed relatively and not active. So this kind of differences between the genome sequences can be obtained. Using the technique, another NGS-based technique called Hi-C or CHIA-PET. This technique also uses the NGS, the DNA sequencing, but it can detect the structure of the DNA looping or the higher order chromatin structure. So using this Hi-C technique, the AG Nagai again collected some publicly obtainable data of Hi-C with various cell types, and compared how the distribution of AB compartments are shared or different each other. So here this is the example of the AB compartments. upper peaks shows the position of A compartment, while the lower peak shows the positions of the B compartments. And they are the profiles of the ES cells, B cells, cortex, and sperm. And so you see that the overall, that the AB compartment, the distribution of AB compartments are almost similar each other, but they show some subtle differences. So it is particularly interesting to see if the genes that are located in such a variable regions of the AB compartments are related to the cell type specific function of the cells. So here is an example of such analysis that we compare the expression of the genes, for example, that are located in the regions. Changes the compartments from A to B between the B lymphocytes and the B lymphoma. That is, that if the B lymphocyte becomes cancer, the B lymphoma, these genes expression will be decreasing. That we can expect, and as you expect here, in the left graph, the expression level decreased if we compare the A to B. So, and if we see the functional characterization of these genes, for example, the apoptotic process is seems to be statistically significant. So maybe this reflect some nature of cells, the changes of the cancer formation of the B lymphoma. Of course, it is the statistical significance may not be so high, but we believe that this kind of approach will also be important to understanding how the non-coding regions of the genome regulates the cell type specific expression of genes. So that is the end of my talk. In summarizing my talk, first, the NGS is not only used for just reading the DNA sequences, but also for getting various types of information, such as the higher order chromatin structure, etc. The second, classical rule-based approaches may be suited for the interpretation of genomic information. As I explained, maybe the knowledge-based, rule-based systems would be the system that nowadays all people are studying deep learning, etc. But I think that the more classical rule-based approaches would be also important, become important again to characterize the genome sequence, because the size of the genome is limited. Finally, a remaining big question is how the epigenetic information is indicated by the genetic information. The unification, the integration of the first approach I explained, and the second approach I explained, that how to integrate these kind of approaches would be the remaining question. That's all. Thank you for your Thank you for your attention. I will accept questions if time.
Sir, thank you so much for your valuable presentation, sir. And thank you for your speech on machine learning approach, and also one genome, many cell type approach. And also you have given your enlightened talk on new technology called single cell RNA sequencer. And very much thank you for your valuable time, sir. And session is hand over to Akila.
Thank you, sir, for your wonderful words. Moving on, I'd like to present to you all the profile of Dr. G. Narahari Shastri. Dr. Shastri is currently working as the Director of CSIR, Northeast Institute of Science and Technology, Jorhat. Dr. Shastri is a chemist working in the interdisciplinary areas spanning chemistry, biology, modeling, and informatics. He was awarded with Shanti Swarup Bhatnagar Prize in Chemical Sciences in 2011, considered as one of the highest prize for science and engineering in India, National Bioscience Award, TBT 2009, one of the highest for life sciences in India, Swarna Jyoti Fellowship 2005, B.M. Birla Award for 2001, B.C. Death Memorial Award 2009, CRSI Medal 2011, and Alexander von Humboldt Fellowship. He is also Sir J.C. Bose National Fellow. He has delivered more than 350 invited lectures, which include talks in national and international conferences. He was a visiting professor at IMS lectures, which include talks in national and international conferences. He was a professor at IMS Japan, LMU, Munich, Germany, Jackson State University, USA, and Kyushu University, Japan. He is a fellow of National Academy of Sciences, fellow of the Indian Academy of Sciences, fellow of Royal Society of Chemistry, fellow of Association Biotechnology and Pharmacy, Telangana State Academy of Sciences, and Andhra Pradesh Academy of Sciences. He was recently elected as a fellow of INSA and thus becoming a fellow of 3 most prominent science academies in India. He has more than 282 publications with over 10,300 citations and an H-index of 51. He has successfully guided 24 PhD students and 15 postdoctoral fellows, and currently about 10 researchers are working as postdoctoral fellows for PhD students. Dr. Shastri's research interests are theoretical and computational chemistry, computational biology, computer-aided molecular design, chemoinformatics, and big data and artificial intelligence. Dr. Shastri has made fundamental contributions in the area of non-covalent interactions and developed several important concepts in this area. His group is interested to apply the data science approaches and also developing indigenous software, molecular property diagnostic suite. He is a regular reviewer of several journals and also on the editorial advisory boards of some of the journals. Sir, it's a matter of immense pride to have your presence amongst us today. May I now take the privilege of inviting Dr. J. Narahari Shastri to please speak a few words. Sir, please.
Thank you very much, Akhila. At the outset, I would like to thank Dr. Rangarao and Dr. Kripanidhi, the Dean of Planning and Monitoring, and definitely Dr. M.Y.S. Prasad Garu and all the management of Vigyan for giving me this platform to share some of the work that we are doing. Before that, I also would like to thank my good friend, Dr. Prashant Suravajula, who is one of the force behind BioClues and also this AP Bionet, who was responsible for me to accept this invitation and then give a talk. I would like to now share my slides. Today, I'm going to talk about, I'm trying to give a minute. Are you able to see? Can somebody say yes?
Yes, sir.
Yes, okay,
yes,
thank
sir.
you very
We're
much.
able to see.
Okay. Thank you. And I would like to give a overall view about the infection and immunity in the context of the current pandemic that is going on and the role of modeling and informatics. So if you look at drug discovery, as this is one of the first talks, and after hearing Professor Kenta Nakai's talk, I thought that we will also have an introduction about drug discovery. And in drug discovery, about 200 years before, it's all about some vaccines or some serums, and small compounds were used to treat diseases. And this also went on for several years, where the small molecules are drugs And there was a concept called lock and key mechanism, where a small key will interact with a biological receptor, which is a lock. And that also led to different types of hypotheses that are there in drug discovery and gave rise to pharmaceutical sciences and medicinal chemistry in a very big way. And then rational drug design aspects have started based on the structure-based approaches and analog-based approaches, where basically an analog-based approach is you have a key which might open an unknown lock. You don't know the mechanism of action, but you know that this particular small molecule will have an efficacy against a particular disease. And in contrast, if you know what is the receptor, you would like to make a suitable key so that the particular lock can be targeted, like different inhibitors, which will further become drugs. Afterwards, there are monoclonal antibodies and intracellular inhibitors and epigenetic drugs and oligonucleotide therapeutics, and the most important thing is genome editing with CRISPR-Cas and next generation sequencing, as it has been discussed in the earlier lecture, and network therapeutics, and then personalized therapeutics, because even when different people have the same disease, the manifestation of the disease in different people will depend on their genetic makeup. So the personalized medicine is going to be a big thing. So we have a slow transformation of the paradigms in drug discovery, from rational drug design to intelligent machines, and artificial intelligence and machine learning and deep learning are playing a very, very important role. So slowly we are going from target-based therapeutics, which is very, very definitive and simple, to molecular mechanisms, to phenotype-based drug discovery. I just wanted to tell you that the drug discovery protocols can be based on a disease type or from a drug, or you can have, this is a very cluttered slide, and I don't want to spend a lot of time in this. I just wanted to tell you that there are different approaches for drug discovery. One is a drug-oriented approach, and another is a disease-oriented approach, which is mostly based on the knowledge-based and experimental approaches, which gave some clues, and then we go ahead and then try to develop drugs based on these clues. When molecular modeling and computational approaches played a very important role, the structure-based and ligand-based approaches have played a very strong role in drug discovery. And the drug discovery is a highly multidisciplinary field which involves a large number of people. So there are two distinct verticals which are coming up. One is discovering a new drug or a new chemical entity, essentially a small molecule which will go through the preclinical trials and clinical trials and get an approval from the regulatory agency. And worldwide, the regulatory agency is Food and Drug Administration of the United States, and each country will have their own regulatory agencies. So they will clear it, and then it can be used in the population. The other important development that is there is it possible for us to use a drug which is which has an approval against a particular disease? Can that be used for another disease? This is drug repurposing or drug repositioning, which is also playing a very, very important role. And this particular making new compounds or repurposing the known drugs can also be applicable to vaccines. And in our country, recently, the other approaches like phytopharmaceuticals, which will be a mixture of compounds, that is also picking up. It is not a new chemical entity, but a phytopharmaceutical route to find a therapeutic for a disease That is also coming up. And another route is an Ayush route, where we will try to get the traditional wisdom from Ayush. There is actually an MOU which is signed with CSIR and Ayush Ministry, where one can take the mixtures or the therapeutic procedures which were there in our tradition to be scientifically validated, and there is a possibility to get the regulatory approach. So fortunately, on this particular Ayush mode, we don't have to go through the preclinical studies, and then it can be used based on the clinical trials. So if you look at a well-known strategy, that there is a disease, and there are several genes associated pathways that are there which can be related to a disease, and this can be related to some metabolic pathways, and from there you can get some druggable targets, and one can have a drug with one or more targets which indirectly or directly associate with the disease. And let me try to tell that, okay, disease is there, and how do you define a disease? I will try to take some time to put some simple things in perspective, that things like sickness, illness, malady, and disease are sort of not very clearly understood. And according to WHO, the definition of health is a complete physical, mental, and social well-being, and not merely the absence of a disease or infirmity. We'll also try to look at what is infirmity or frailty in a more quantitative fashion. And disease in some way can be treated as a perturbation to health. If a person has an ideal health, and if there is a problem with that person, either in physical or a mental or a social dimension, then probably the person is not healthy. If the person is not healthy, maybe there is some disease which can be associated. So there is a strong relationship between immunity, infection, disease, malnutrition, and inadequate dietary intake. And there are other social aspects which are also there, and we cannot really ignore. My idea to kickstart in the beginning of this seminar is to bring to the notice of all the audience that what we are talking in terms of developing a drug or the current year's focus of InSyB 2020 is bioinformatics for disease therapeutics. We will have to look at what are all the factors that are involved in disease therapeutics. So this infection and immunity will be very important aspects, and we have the innate immunity and adaptive immunity. And the innate immunity, there are different pathways like cytokines, dendritic cells, and chemokines, and several very well quantified pathways are there which are related to innate immunity. And there are also several aspects for the adaptive immunity. And I would like to tell that abnormality or dysfunction of the immune system may cause an unexpected immune response, as seen in the case of hypersensitivity, autoimmunity, or immunodeficiency. Autoimmune diseases and disorders are really very, very dominant in many cases, and immunodeficiency is probably one of the strong reasons for people to be infected with infections. And one of the major things that is going on now is the COVID infection. So immunomodulation is key. And even for cancer, after people looking at chemotherapy, radiation, and surgical intervention, the immunocheckpoint therapy seem to be play a very important role. And it's very interesting to look at the infection or in some way allergy. and some way the personalized medicine. These are very, very interesting aspects when you want to understand the pathophysiology of these diseases and the manifestation of these diseases in general and manifestation of these diseases differently in different populations. So if you look at the virus, the family, morphology, and transmission, you have different types of viruses that are there, and DNA viruses, RNA viruses, and retroviruses. And the understanding of these viruses is becoming more and more due to strong progress that is made in structural biology and protein crystallography, cryo-electron microscopy, and we have a lot more quantitative information about all these DNA viruses, RNA viruses, and retroviruses. So this coronavirus is an RNA virus, and we have a lot of information on the series of these viruses. And there are many drugs which were approved for these viruses. And it's also important to understand the mechanism of these things and then how effective they are. So it's a strong combination of medicinal chemistry, structural biology, understanding of immunity, and understanding the whole metabolic pathways, which involve a lot of understanding of genomics, proteomics, and if you go to the personalized medicine, there are many, many other aspects which are involved. So if you look at the host-pathogen interaction, understanding the molecular basis by which pathogens interact with human and plant hosts, with the ultimate goal of identifying targets for prophylactic or therapeutic treatment. This is very, very important. Why and how a particular pathogen will interact or will not interact with humans. These host pathogen interactions are very important, and there are quite a few people who are working on this for a long time, and because of this post-COVID, there is a lot of interest in CSR labs, and we are trying to work with many labs, with the THSTI, which is a DBT lab in Delhi, IBST, IIT Guwahati, along with other DRDO and other labs also, to understand what are these host-pathogen interactions, their origin, their molecular pathogenicity, and if possible, can we have the virulence markers, models, biomarkers, therapeutic targets, and diagnostic development. And this, if possible, can we translate them into therapeutics? So if you see the host-pathogen interaction, the most important thing is how these spike proteins will be interacting with the host, and we can look at different potential drug targeting spike proteins, like we can have neutralizing antibodies, or we can also look at fusion inhibitors, or we can also look at host protease inhibitors. And in all these three directions, a lot of work is going on in India and abroad, and hopefully our understanding of this virus and host pathogen interaction will lead us to have a clarity on our immunity and inflammation. As I said, the WHO definition of health is a state of complete physical, mental, and social well-being
Akila, is everything fine there?
Yes, sir. It is everything fine over here.
Maybe our systems are going well. I think problem with SAR only.
Yes, sir. Yes, sir.
Wait for some time.
Yes, sir.
Please.
We'll wait for some more time.
Yeah, please wait.
All right, sir.
Anil.
Yeah, I can hear you, sir.
Yeah, the problem is with Sara.
It's the problem is for SARnet only, sir. Our system is fine. Sir, sir has just joined now.
So I'm so sorry that I don't know at what time I went off. There was a.
Yes, sir. Thank you.
At this. So this I have shared it, right?
Sir, please
Are
share
you
your
able to
screen.
see my slides?
No, sir. Please share your screen.
Are you able to see this?
No, sir. Yeah, yes, now we can see you, sir.
So now you also have to tell me when did I stop?
Yeah, could you move further and further? I will let you know, sir.
So this is over. This is
Done.
over probably.
Done, sir.
This one is over. Maybe here.
Yes, sir. From this slide onwards.
So Yeah, I just wanted to tell you that this health is a state of complete physical, mental, and social well-being, and not merely the absence of disease and infirmity, which is the definition given by WHO for World Health Organization. So there are aspects like pain, frailty, aging, inflammation, infection, metabolic and genetic disorders, and exercise and lifestyle as physical fitness, which we can talk about physical, but there are mental and social aspects which also need to be considered. So if you want to really look at the physical and a little bit mental aspects, so you can see nutrition, exercise, sleep, and lifestyle, and in general weight management or eating habits are one of the factors. And you have mental and social issues which are there, and balancing the pressures and materialistic pleasures that we have is also very important. So there is a lot of scope to quantify these things by using computational approaches, whether it is data science, machine learning, or it is also structural biology or molecular modeling. So earlier, we have this one disease and one target approach, but you have one target and multiple drugs, and multiple drugs and one target are also possible, which gave rise to network pharmacology and systems biology aspects, which are very important for all of you to consider when you are looking at bioinformatics, especially in drug discovery. So I also wanted to bring some challenges in drug discovery, that not only getting a molecule which is having a therapeutic potential is important, but there are aspects like delivery route. What type of a delivery route that one has, oral or perinatural, and also one has to look at drug-drug interactions, differentiation from current therapies, dosing frequency, what is the cost of the drug, what is the risk of misuse of these controlled substances, what are the side effects, and what is the safety in some selected population, and the dose adjustments in pediatric patients, and the monitoring and the specialist use of these things are also very, very important when you are trying to develop a drug. And I just wanted to tell you that disease interactions is very, very important. So when somebody is having a disease or using a particular drug, and when a new disease comes, how will the new disease be manifested with an individual who is already having a particular disease is an extremely important aspect. And in very simple words, if somebody is having a disease and they are taking a medication, for example, if somebody is having a diabetes, and if they have another disease like a COVID infection, how do we treat that particular person may be a bit different compared to a person who is normal and then have a COVID infection. And you can easily say that somebody who is a woman who is pregnant, they may be different population compared to normal people. Like that, so there is a lot of work that has to be done. So what we have done in our group is that there are a large number of ways in which a particular disease is manifested. And if you want to develop a drug, you will have to consider these multifarious aspects and have a platform or a web portal or a software which will be able to bring all these tools in one place. One of the biggest problems that we have in academics is, while a large part of the bioinformatics software for functional use and databases are free, thanks to EMBL and other community, the computational drug discovery softwares are usually very expensive, and it is sometimes beyond the reach of the general people. So there are two problems here. One is, it is very expensive to buy And the second thing is all of them are coming as finished products, and then it is not possible for somebody to contribute their knowledge into this. So we would like to have a platform wherein people can freely use the software that is available, and also if they write a small code which can be incorporated, and this is implemented as molecular property diagnostic suite. And I have started this about 8-9 years before in Hyderabad, and when I moved to Jorhat, we're also working on this. So it essentially have different modules like data library, data processing, data analysis, and advanced modules. And I just wanted to tell you that the data library will contain all the information about a particular disease and the targets, the literature, the gene library, and the compound library. Data processing will have the file format converter and descriptor calculations. And data analysis will have molecular docking, QSAR, virtual screening, drug repurposing, visualization, sequence analysis, and active site preparation. And advanced modules will have some machine learning tools, text mining, and other computationally intensive tools. And we also have some very specific thing about the disease mechanism. And these modules can be divided into disease-dependent modules and disease-independent modules. And we have first started that with tuberculosis. And now with diabetes and metabolic disorders, they are completed and we have published. And currently COVID-19 portal is coming up, and HIV and other portals are coming. So the workflow, we have data library, data processing, data analysis units, and then you can probably refer to the publications that we have. And I just realized that we are running out of time. Dr. Rangarao or somebody, can you tell me that how much time you will give me to wrap up?
So try
because
to
I have a problem that if you don't
minutes,
control me,
yeah, try to
another 5 minutes.
another 5 minutes.
Okay, okay, thank you.
Yeah, thank you.
So what we have done is we try to give a number for every single compound in the chemical space. And I am very happy that this particular exercise is done in the world for the first time in our group, where we have taken all chemical space into different classes and clusters, and each compound will have a unique ID that is very much similar to an Aadhaar card. And I feel that the molecule is like a human being. So as every person in India has a Aadhaar card, we would like to give an Aadhaar card for each molecule, which will give some properties of this in the first page, but it might have second page and third page, and we are constructing. And so far, we are able to create this database of 130 million compounds. And this particular data will be very, very useful in subjecting them to a large number of machine learning and AI and other data science techniques. So these MPDS portals are coming up, and I wanted people to use them. And I will just go through very quickly. This COVID portal is also coming up. I will just quickly talk about our institute work that we have done. And we have done the COVID testing. And so far, this number is about 38,000 samples we have done. Now we are able to do a lot of samples, but we are not getting that many samples, though. That's why we are not doing it. Otherwise, we have started this COVID testing, and also we have done the serological study, zero time from a Phenome India, and all the CSIR lab employees were a cohort group, and then this is going to come up in a big way, and then maybe all the CSIR labs, we are about to publish this paper. And another thing that we have done is a summer research training program in CSIR NIST, where we are able to provide training for 16,000 people who have applied within a gap of 10 to 15 days. All the applications have come, and then we started this training from July to September in this year, and where people like N.R. Narayan Murthy of Infosys and Arya Mashelkar and the PSA to government, Dr. Vijay Raghavan, and a large number of people have come. And then this was a very good experience, and we have more than 1 million visitors for our SRTP website. And we also have conducted some drug discovery hackathon program, which might be relevant. I wanted many of you to visit this website, which is available. Many lectures are available. In 3 verticals we have conducted from July 13th to September 11th. And vertical 1 is focused on theory and computational methods. And vertical 2 is focused on algorithm and method development. And vertical 3 is based on open source and commercial software. All the commercial software people like Schrodinger, BioVia, Chemexon, Cresset, they have come and then given the demonstration. And everything is done online, and the CSIR East was the platform. And this is all possible because of the enthusiastic participation of many of the people. This is a group photo that we have done with a bioinformatics workshop. And thank you very much. And I'm very happy that I'm able to finish the lecture despite our internet problems. Hopefully you are able to listen to this lecture through online. Thank you very much.
All right, sir. That was informative and intellectual too. Thank you for delivering your insights today despite your busy schedule on this occasion. Thank you very much, sir. Now let me take the pleasure to invite Dr. Abhinav Parashar, Department of Biotechnology, Vignan's Foundation for Science, Technology and Research, to introduce the president of this session. Sir, please. Sir, could you please unmute yourself?
Sorry. Thank you, Akila. I hope I'm audible now.
Yes, sir, you are audible.
Okay, great. Okay. Dr. Laurataya, founder and chief of Vignan Group of Educational Institutions under the aegis of Love Educational Trust, established the flagship institute, Vignan's Foundation for Science, Technology and Research, almost a decade ago in the year of 2009. He has always dreamt of making the institute into a center of excellence through collaborative research and industry with industries and premier institutes of India as well as abroad. Dr. Rataya is an MSc in chemistry, and he obtained his PhD in educational psychology from Osmania University. He's a great visionary and a very well-known figure in Andhra Pradesh, and has been quite instrumental in affecting several educational reforms by serving as an advisor to the Andhra government, not once, multiple times, multiple instances. Being alarmed from the fact that many rural youths dropped out from colleges and schools while he himself was a student, decided him to educate at least 10 people. So slowly things became feasible, and once it was feasible, he started the first institution in 1980. And today, Vignan's group has a commendable presence at all levels of education, with 16 schools, 8 junior colleges, 8 engineering colleges, 3 pharmacy, and 1 university, which is educating more than 40,000 students. His dynamic personality in channelizing student energies and motivating them towards higher intellectual pursuits right from their early teens. Thousands of successful engineers, doctors, lawyers, technocrats, and entrepreneurs acknowledge Dr. Rataya as their mentor. I would now request Rataya sir to kindly share his insights on InSyB. Sir, please.
Okay, thank you, Abhinav sir, for giving me a great introduction of myself. So thank you very much, which I don't deserve this kind of introduction. Okay, anyway, as the chairman of this Vignan University and Vignan Group, I am very happy to learn that our biotechnology department and bioinformatics department is conducting a symposium on therapeutic and also diagnosis part. Yes, that's with the support of the whole administration of the university, from Vice Chancellor, and deans and everybody. I really congratulate the departments of biotechnology and bioinformatics for this kind of, for the organizing this kind of symposium. Thank you and congratulations. And I'm more happy that this symposium involves many scientists, many academicians, and I just listened to Narsamurthy and other people. So this kind of great people, they are involved into this symposium. That is the greatest thing. Just not only just mere conduct of symposium namesake, involving many people of different institutions of at the different levels, higher levels. This is the more important, and this is the most happy part from my side and from the institution side. I really, as the chairman, I welcome all these external scientists and academicians who are involved into this. So I welcome Dr. Muhammad Asif Khan, Turkey, Dr. Kenta Nekai, Japan, Professor G. Narhari Sastry, Director, Northeast Institute of Science and Technology, Professor Bruno Geta, Australia, Professor Ramaswamy, Singapore, Professor Y. Kit M, Malaysia, Professor GPS Raghava, IIIT Delhi, and Dr. Prashant, Birla Institute of Technology and Science, Jaipur, Dr. Tikram Chand, Sukhadia University, Udaipur, Dr. Sugunakar, Lovely Professional University, Ludhiana, Aparna Banerjee, Chile. So I welcome all these people who are involved into this symposium, and it enriches the whole environment, I'm sure. Yes. And also I congratulate our departments and also the participants for taking up a very good topic, that is the futuristic topic. The future is, as all of you aware, so most of the doctors may be replaced or supported by AI doctors, artificial intelligence doctors. And most of the drugs are evolved out of the machine learning or artificial intelligence. And the future is, yes, like Aadhaar card, another DNA card may be given for every individual. And the total diagnosis and the treatment goes personalized against the present situation of the general diagnosis. Suppose if we get the headache, everybody is given an acid kind of thing. Now, once this DNA and other machine learning and artificial intelligence is into this, definitely 5 people will be given 5 kinds of treatment depending upon their DNA and other information. So this is where the bioinformatics will be into this. And the future students, scientists, and teachers have to evolve themselves. And biotechnology people, really biotechnology people, if they are limit themselves to their present knowledge, they may be irrelevant within few years. Yes, so they have to update themselves with the knowledge of the emerging trends like machine learning, data analysis, and artificial intelligence. Otherwise, they would be irrelevant. Even in biotechnology courses and other related courses also, if you don't introduce these tools, I think it may not be useful for the future. Yes, that's my, a perception, my personal perception about the future. Because it has lots of advantages, involving, introducing the AI or machine learning into this therapeutic and diagnosis process. Because so now the present doctors are not connected to one another. Naturally, maybe if there is a connection, the connection is very limited. Once it is AI doctors, they are connected. That is the biggest advantage. All the computers are connected world over. The second thing is, so information is very fast. Suppose somebody discovers a drug, So to popularize the drug, it takes minimum of 2-3 years to spread into the all sections and all levels of the society. But once the machine learning is introduced into that, AI is introduced into that. So the popularity of the drug today, if a fellow knows it in New York, we'll know it the same day about the impact of the drug or the properties of the drug. So that is it, what is going to happen. So the professors, all of you are real scientists. I am not a scientist. So I am not here to teach or preach anything, but this is my perception. As the chairman, I am expressing my perceptions. That's it. This is the future, I believe. So from the last 10 years, sometimes the perceptions may be true. So from the last 10 years, I'm repeatedly telling water would be the biggest commodity. Recently, an American firm has introduced water future. Yes, they're selling it, the water. They are quoting the water now and selling it at a later stage like petrol and other commodities. Same way, yes, this in the biotechnology field also, yes, that became a very big company, Water Future. You can go to their website and see how the water becomes the commodity. Yes, it is estimated after 10 years, water may be scarce in some of the countries. So they made it a commodity. They are selling it now. They are quoting the price now, and they deliver it at the appropriate time. This is it. What is going on. In the same way, in this field also, the way to being scientists or being the guides for many researchers and students, you should look about the future, not thinking of the present. The future is interference of AI, interference of machine learning, interference of data analysis, and all the other things, other knowledge parts into this biotechnology. And of course, bioinformatics is evolved, but it needs to be speed up. It needs to be it needs to be faster than the present moment. Anyway, our faculty members have done. Worked out on corona and other diseases to some extent, and they published some papers also. I think in these 2 days they'll tell the details, but they should go in a bit faster way with more samples. As all of you heard, that in UK the virus has taken a different pattern, different shape. So this is how we have to. Nobody could, everybody is trying for the drug, but nobody is trying to map the genome of the virus. And of course, it may take some more time, but this is how it should go. Why this much? 1 year they have taken the time to map the genome of the virus, corona. So that is a very sad thing. So we are still going slow. So being scientists of this area, of this area of knowledge, so try to make it faster and suggest things and to go still in a speedy way. And it will be greatly help for the whole humanity, not this country, other country, but once you involve this technology or information and the tools of different tools of IT into this, it would be a beautiful thing. I hope, I wish this symposium will throw some light on towards this direction. I'm once again happy that many countries have participated into this symposium. I think some of you must be doing a big work and a good work also. And if it is shared, I think the other people also get benefited. In fact, we get our university and our biotechnology and information department, bioinformatics department will get a lot of benefit out of this symposium. I once again thank all the participants and all the contributors, resource persons, for sparing their valuable time and to enrich the other people. Actually, knowledge always, I believe, knowledge always grows by sharing, not simply, making it confidential or making it secret. Knowledge always grow by sharing. And this is the kind of sharing of the symposium is also a kind of sharing of the knowledge. So I strongly believe that this would be a great benefit, especially for our departments of biotechnology and bioinformatics, and all other people also who came from Malaysia and other countries also. And I hope and I wish that there are many senior scientists who have participated in this seminar, in this symposium, and they will throw more light how to go faster. We have taken already 10 months time to map the genome of the corona, not yet made perfect. How to make it faster? How to involve the how to design a speedy process. So that will make some big sense. So for India, yes, if you are able to do the Aadhaar card, definitely we can do this. Another health card with the DNA structures, mapping and all those things for every individual. And that will give a lot of relief for the poorer sections. Yes, instead of now, I think all of you are aware that everybody without the diagnosis, without any medical support or doctor's support, many people spend more than 5 to 10 lakhs for the treatment of corona without any treatment. That is underlying. No treatment is there because it is not diagnosed. No medicine is there, so they can't do it. No doctor is specialized in this, but they have charged from 5 lakhs to 20, 30, 50 lakhs of rupees. Yes, nobody knows why they charged. Nobody knows how they have cured. Nobody knows what drug it has helped. That is the very, very, very sad part, because nothing is analyzed. Not only here, in the entire world, nothing is analyzed. Why the particular drug is useful or helpful, why a particular line of treatment protocol is useful, nobody knows. So that is the saddest part. Who has to do this? You people have to do this, not the doctors. You biochemists or biotechnology people or bioinformatics people have to do something, throw some light on this. So maybe we are expecting some more versions of corona or different versions of different viruses. So we must gear up ourselves to have very speedy solutions for all these things. Once again, I thank you very much for your participation. I thank the participant students, scholars, and other students also. I heard that more than 900 students or scholars have participated in this symposium. I am very, very happy that definitely they will be enriched with all these, by listening all these talks. Thank you very much for giving me this opportunity. Thank you, Abhinav. Thank you.
Thank you very much, sir. Thank you for your valuable words. That was very much intuitive. Now, let me take the pleasure to invite our Honorable Chairman, Dr. Lavrataya Garu, to release a book which was edited by Professor P.B. Kavakishore, entitled Genetically Modified Crops, Current Status, Prospects and Challenges, Volume 1. Sir, I would like to request you to release the book. Sir, please.
Yes.
Thank you.
This is the book written by our Professor Kavakishore. Professor Kavakishore is working with us as an advisor, and he is contributing a lot for the development of the biotechnology department in terms of good number of publications and applying the funded projects in many ways. Kavkishore is doing in a very committed way. I really congratulate Kavkishore for authoring this book. So, genetically modified crops. This is the title of the book, Genetically Modified Crops. So, current status, prospectus, and challenges. This is volume 1, published by Springer. And I heard that volume 2 will also be released within a week or 10 days. I really congratulate Kavkishore Garu for this kind of contribution, a great contribution. Thank you. Congratulations to Kavkishore Garu. And thank you. Thank you very much. Okay. Thank you all. Thank you. Okay.
Thank you very much, sir. Stepping further ahead, I would now like to present before you all the profile of Dr. Bruno Gaeta. Bruno Gaeta is the Senior Lecturer and Director of Studies of Bioinformatics Engineering Program in the School of Computer Science and Engineering at the University of South Wales in Sydney, Australia. He trained as a molecular biologist before making the switch to bioinformatics, before it became fashionable, and worked in both academia and industry as a founding member of Australia's first bioinformatics startup. His research interests span the study of sequences, structures, and protein expressions with focus on the applications of computational biology to understanding the immune system. He is also a passionate advocate of the place of bioinformatics in scientific discovery and education. He is highly active in professional societies as treasurer, executive committee member, and director of the International Society for Computational Biology, and has been an executive committee member of the Asia Pacific Bioinformatics Network since 2008. Sir, it indeed is a matter of immense pleasure to have you amongst us today. I would now like to request Dr. Bruno Gaeta to please speak a few words. Sir, please.
Thank you very much. I will share my screen and start the, let me make sure which one it is. No, Zoom wants, I just updated my operating system. And okay, I'm going to need to quit and restart because Zoom wants me to do that. I'll be there shortly.
మీకు వర్షన్ కొందరు చాట్లో కామెంట్స్ వచ్చినాయి పక్కన చాట్
Hello again, let me share my screen again. Okay. So, first of all, I would like to thank the organizers for inviting me to speak at this symposium. My talk is going to focus on immunoglobulins, better known as antibodies. Now, antibodies have been the focus of an enormous amount of work. It really is something that impacts medicine through and through. And of late, we've heard a lot about antibodies due to the pandemic. So just to remind you that antibodies are those Y-shaped molecules that interact with viruses. This slide is from the PDB database, and it shows actually the rhinovirus, common cold virus, to scale with the antibody here. This is one example of an antibody. This is an IgG. And the antibody is this Y-shaped molecule, which has got these variable regions. And those variable regions bind antigens. They bind foreign bodies and essentially prime them for being recognized by the immune system, for being destroyed by macrophages, and so on. And really, the antibodies are what constitute the memory of the immune system. The antibodies, the variable regions, emerged through this accelerated evolutionary process, this combination of mutation and selection in the variable regions, which means that there's a huge variation in the variable region of the antibody. And it provides highly specific binding and recognition of a specific antigen. So what I'm going to talk about is a range of work that we did over the years focusing on antibodies, starting from talking about antibody genes, immunoglobulin genes. These are the genes that encode the antibodies and are really responsible for the variability in the antibodies. And if time permits, I'll speak a little bit about the work that we're doing now on the antibody protein, always using bioinformatics as a way to try to understand these molecules and how they work. So starting with antibody genes, obviously there are millions and billions of possible antibodies, but these are not all encoded in the genome. What is encoded in the genome are those germline genes that represent parts of the antibody sequence. And when the antibody producing cells differentiate, those genes get rearranged, and you get essentially 1V, 1D, 1J gene that gets spliced together to form a specific novel antibody variable region. And this process is different in every antibody producing cells, which means that as those antibody producing cells differentiate, they all result in different immunoglobulins. But that represents only part of the diversity story. This is only one mechanism through the diversity of antibodies is introduced, because this joining that happens between the V, the D, and the J is actually an imprecise joining. And there's a range of processes that happen that I'm not going to go into, which are P addition, N addition, there's action of isonuclease that removes nucleotides. And then there is the process of hypermutation. And the process of hypermutation is what leads to the refinement of the antibody so that it matches an antigen really precisely. The somatic hypermutation means that when the immune system encounters a new antigen, those antibodies that match this antigen get encouraged to divide, but also to mutate. And as they mutate, some of those antibodies are going to match the antigen better. And those that match the antigen better mutate further, so that what you have is this accelerated evolution process that ends up with the production of an antibody that matches the antigen very closely. And this is how the immune system develops some form of memory. Now that process takes a bit of time to happen. And it's dependent, it does happen that the antibodies are always going to converge towards an antibody that matches the antigen, but it can take more or less time depending on what you start with. And what you start with really is the antibody genes that you have. So there's also a lot of interest in those germline genes. So the germline genes is what inherited from your parents. These are those component genes, this V, this D, this J gene. So there is a lot of variability in those genes as well. So there is a lot of individual variation between people as their antibody genes. But that is very difficult to characterize. Pretty much all the projects that have focused on variation in this have ignored this locus because it's too variable, it's too confusing, and often the human DNA is sequenced from rearranged antibody cells. So you actually miss out on this part of the genome. And because it's a region which is very repetitive, it's very difficult to sequence through as well. Although it's changing now that we've got technologies such as nanopore. It's just showing you a quick picture of that locus, which is very complex. But this is what you look like if you look at it in the 1,000 Genome Projects. Essentially, this entire region is masked with a big deletion. So the way in which myself and my collaborators have approached that in the past is to look at mass sequencing of rearranged immunoglobulin gene sequences. So instead of trying to sequence the germline genome, we sequence the antibody producing cells. But of course, every one of those is going to be different. So what we get here by sequencing these variable regions is a very large number of sequences. And we started doing that with 454 sequencing when it started. And now a lot of other technologies can take the place of that. But the process here is once you have these data sets, which can have millions of different rearranged immunoglobulin genes, how do you work your way back to the germline? And based on these data sets, we really developed a series of methods that allowed us to identify clonally related sequences, to define the genotype and to define the haplotype of an individual based on those rearranged sequences, as well as refining the germline repertoire. And what's core to all of these methods is an algorithm, a model that we developed early on, where what we called Immunoline. It's spelled that way because the HMM there indicates the fact that this model is based on a hidden Markov model. Now, if you have done bioinformatics, you have heard of hidden Markov models. They are essentially a probabilistic model of a process. And in this case, we have built this hidden Markov model to represent this process of antibody generation, of gene joining, rearrangement, mutation, and so on. And that model is a probabilistic model. It's informed from known data about the rearrangement of immunoglobulin sequences, as well as the information which is present in the sequences themselves. And the goal of this model originally was to say, okay, here we have a rearranged sequence. Can we predict its component germline genes? And what probability can we get from that? So without going into the details, this model reverse engineers a series of steps in the immunoglobulin generation. The first step that it models is the immunoglobulin rearrangement process itself, so the splicing of those genes. And then the model incorporates stages that model all these different processes. So the P addition, the addition of palindromic nucleotides at the junctions, and addition, which is a non-template encoded addition of nucleotides, exonuclease action that actually removes nucleotides, deletes nucleotides at the junction, as well as the process of somatic hypermutation. And it does that by modeling the position dependence of the somatic hypermutation. So the probability of mutation depends on the position along the sequence, for example. And it also depends on the sequence of the that's being mutated. So the traditional model there is that the sequences, those consensus sequences, RG, YW, WRCY, WAN are hotspots for mutation, and they're more likely to be mutated by this process of somatic hypermutation. So my colleague Andrew Collins spent quite a lot of time refining those models and developed, in fact, a trinucleotide score model, which is incorporated in Immunoline. So Immunoline pretty much originally works as a way to identify the germline genes, starting with a blast search of the V gene, which is long enough to be identified with a blast search. And then it builds the HMM based on prior knowledge and based on this alignment. And once you've got the HMM, then you can take an individual rearranged sequence and feed it through the HMM and reconstruct its origin, reconstruct its germline genes. This is just showing what the HMM looks like in terms of topology. I won't go into the details there, but suffice to say that this HMM works pretty well at reconstructing the germline gene history of a rearranged sequence. We developed, as well as Immunoline, we developed a number of benchmarks to evaluate the performance of the system. We use clonally related sequences. We use randomly selected sequences. What we found is that Immunoline did better than the other programs that try to do the same thing. All of those are details in our papers. We also developed a data set that is used to test those different methods. And again, Immunoline overall has the best performance across the all three gene types, V, D, and J. Some methods have a better performance with, by the way, shorter bars is better here. Some of them have better performances with one of them, but overall Immunoline is better. Now, Immunoline was a long time ago, and it's no longer being developed. There's been other methods that have come, newer methods that have now replaced it, but all these methods actually have adopted the same algorithm as Immunoline. So the thinking behind Immunoline underlies most of the modern methods now. But this model, which is more than an alignment method, it's a probabilistic model, helped us to also look at identifying clonally related sequences. So when we sequence these millions of rearranged immunoglobulins from a donor, from an individual, what we want to do is be able to reconstruct their history, to reconstruct it in the form of trees that way. So one of the approaches that we used was to try to identify those groups of clonally related immunoglobulins, pretty much by using a clustering approach. So using Immunoline, we were able to develop this a way to build a distance matrix from pairs of sequences, and from there you can cluster them, build a dendrogram, and then select a threshold, and that threshold allows you to identify clusters pretty much. And that provided a method to identify those clonally related sets. I'll skip the slides. One very interesting problem that we then faced is to try to reconstruct the germline genes of the individual. So, as I mentioned before, it's very difficult to sequence the genome, the germline genome, in the immunoglobulin locus. But it's very easy to sequence lots and lots and lots of rearranged genes. So the question that we asked is, if we know the sequences of all these rearranged genes, can we work out the germline genotype, which V, D, and J genes that individual has? And that's pretty much trying to identify the combination of alleles that maximizes the probability of the observed data. The method that we have uses a series of steps. And when we tested it with different clones and different individuals, it did allow recreating the identifying the genotype. So this particular experiment makes use of data from twins, and you can see that it groups the twins together, as you would expect. So once the genotype is identified, the next step is to determine the, you can then re-identify the germline genes, and you can then refine the process. So it becomes iterative. But then once you've got the genotype, you can also try to determine the haplotype. So which genes sit on the same chromosome next to which other genes. And you can do that again by looking at the rearranged sequences. It's mostly due to the fact that there's a limited number of J genes available. So if you see different V genes that share the same J gene, then they're likely to be on the same chromosome. And again, we address that using a machine learning approach, pretty much a classification approach, trying to classify between a range of methods, of classes. And my student, Gillian Chen, was able to develop this method based on logistic regression that allows classifying the haplotypes. So now I'm just trying to check how I'm doing for time here. So you can also build this utility that allows drawing those haplotypes of the heavy chain variable region locus for different patients. So that's the part of the talk that deals with immunoglobulin genes. When we started this work, we were one of the very few in the world doing it. And since then, it's really exploded, and a lot of groups are now working on this. And it's a very exciting new field. Now all those groups were better funded than us, so we decided that we'd better leave the work to them and move on to really one question that has fascinated me for a long time, which is at the level of not the immunoglobulin gene, but the immunoglobulin protein. The question is really, can we use machine learning to try to predict what antigen an antibody binds to? Because we know, as you've seen, we've got those data of millions of rearranged antibody sequences. So we can sample that from the blood of a patient, but we don't know and we can't determine what these antibodies actually do. We can't tell what it is that they bind to. Unless you go and do the experiment, antibody by antibody, it's very hard to determine. So I'm going to take a project to try to predict, develop methods that can predict the binding antigen based on the antibody. That is, as you can expect, an extremely difficult problem, and we haven't got there yet. What I want to share with you is a couple of approaches that we're working towards. One of them is to try to to use unsupervised learning to cluster antigen into classes, because if we can predict a class, that's easier than to try to predict the individual antigen. And for that, we try to determine, to build a distance metric that represents the difference between binding sites for a protein. Now, that's not a new problem. That's been really done a lot in drug design. There are many different solutions, but they work mostly for very similar molecules. We decided to branch on a method based on pharmacophores. That's something that's really used in drug design. And it's about representing molecules in terms of a combination of spatial and chemical properties, pretty much. But what we wanted was a way to compare pharmacophores and to compare molecules based on their pharmacophores. And what my student Ling Zhao tried was to try to represent these pharmacophores as point clouds. So pretty much take methods that are used in image recognition, especially in 3D image recognition, and try to apply them to our problem here. Now, we only look at, those methods only worked with 3D, 3 dimensions. They're meant to compare objects in 3 dimensions and try to match them in 3 dimensions. But what we're interested in matching are objects in higher dimensions, because we're not only interested in the spatial dimensions, but also the chemical properties. So what Ling Jia did was to try to represent the problem as a higher dimensional space matching problem. And in this case, it chose a couple of, he used a distance matrix designed for pharmacophore comparisons. Pretty much looks like this, where you've got some different scores for different types of chemical groups. And also, and then you have a scoring matrix for the distance between different chemical groups. And then I did some additional dimensions in terms of partial charge and hydrophobicity in the binding sites, and combine that with the spatial locations. So in fact, producing a 5-dimensional space. And when he evaluated that on a number of data sets, some data sets that are used in just traditional binding and also in antibody-antigen binding, and also more general protein-protein interaction data, he found that he was able to recreate, to re-identify the binding from his method, was able to cluster the things that bound the same molecule pretty much together. So quite a positive result. Still needing some work, but promising. But this was really a proof of concept. This is a method. The advantage of it is that it is you can extend it by adding more dimensions. And we haven't really done that yet. The final thing I'd like to mention, I have no idea how I'm going with regard to time. I've just got a couple of other slides here. Just want to mention our current work, which is my student Chao. Our problem with trying to learn antigen-antibody binding is the lack of data. So what Jessica has started doing now is to try to build data sets of antibody-antigen binding so that we can use that for machine learning, pretty much. If we have, hopefully, a data set that's well labeled and is useful, then we can try to start learning binding and hopefully be able to predict what antigen an antibody is going to bind based on the antibody sequence. So what she did was build this data set by using docking between antibodies and antigens, and pretty much build this table. Right now it's still fairly small, but she's used that as a proof of concept to test a range of machine learning methods. And so far, she's getting 76% accuracy in predicting the antigen from the sequence of the antibody, which is promising. Again, we need to get more data and we need to refine this. And this is very much work in progress. But what I've tried to give you in this very accelerated talk was to give you a feel from the sort of work that we've been doing, covering antibodies from the gene to the protein, and really using computational methods to try to refine our understanding and to allow us to make use of next generation sequencing of rearranged sequences, because the clinical applications are huge. You know, you can think of it, you know, if somebody is in the hospital, we can take his blood, we can sequence his antibodies, and we can see what he's got immunity to. You know, that would provide already a huge clinical advantage. Anyway, these are the people who did the work, and especially my students Jillian Chang, Li Zhao and Chao Ye. Mike Bain is a collaborator, and we also collaborated with immunologists and with Stanford Pathology and Defias Group. Thank you very much. I will stop sharing the screen.
Thank you so much, sir, for your valuable words. With this, I would now like to present to you a brief profile of our next speaker, Dr. A. Ramasamy. Dr. A. Ramasamy obtained DPhil and Masters in Applied Statistics from University of Oxford and has an expertise in the area of specialized statistical genetics, bioinformatics, code monkey, and p-value generator. He worked as a research associate in King's College, London, honorary research associate in UCL, Institute of Neurology, and also Imperial College of London. Dr. Ramasamy received various prestigious awards like Young Scientist Sponsorship from European Respiratory Society, the Top Student and Best Student Awards. Dr. Ramasamy is a member in various scientific bodies like American Society of Human Genetics, European Respiratory Society, Royal Statistical Society, and also Science Council. He is actively involved in both teaching and research at various levels. Dr. Ramasamy published more than 85 peer-reviewed research articles with citations 9,000 and H-index 38. Currently, Dr. Ramasamy has been working as the senior scientist at A-STAR, Genome Institute of Singapore.
Hi.
It really is a matter of immense pleasure to have you amongst us today. So may I now request Dr. A. Ramasamy to please deliver his lecture. So please.
Let me first start my video. Hi, everyone. My name is Adai. Don't need to call me Dr. Ramasamy because that's my dad's name. Hi, so can I share my slides? I'll just share on screen one. If I make this big screen.
Yes, sir, we can see your slides.
Can you see my slides, or are you seeing another screen?
We can see your slides, sir.
Okay. Sorry, I got 3 screens. I'm not sure where the hell I'm looking. Okay. Right. Okay. So I just want to quickly, I know this is lunchtime, right? So I don't want to take too much. It's not going to be a very heavy topic. It's quite lighthearted. It's just trying to walk through my career option. I mean, like my career path and see whether some of this is relevant. I mean, I understand there's quite a number of students here, right? So if you all at any moment want to have any, ask any questions, just pop it in the chat or just unmute and just say something. So we can try to make this a little bit more interactive. Is that okay? Right. So first of all, thank you very much to Dr. Rangarao and the Vignan Institute for inviting me to give a talk. It's an honor and privilege to be presenting at the InSyB. I'm just gonna, as I said, I'm just gonna run through a few of my slides. I put the code monkey and p-value as a sort of a joke, by the way, in case you're all wondering what on earth that is. Can you see my second slide now? Oh, you're still seeing my first slide. Okay, somebody's saying chat. Yes, thank you, Nikita. Okay, so what I did for my PhD is basically I was attached to a very prestigious group on clinical trials, and who were very much into study design and how do you get the best study, right? Like because this is the beginning of your experiment. A lot of the omics technology is very expensive. And the last thing you want to do is generate data and then realize it's actually not suitable because your study design was flawed. So we started with a number of techniques, which the first one we did is we try to look for sample size, right? Like we want to know what is the minimum number of sample size we need for a given project. And it turned out that actually at that time, nobody was doing the sample sizes we needed. So rather than just being very negative and saying, no, every study that you have published is underpowered, we changed the topic to say, okay, can we actually combine studies that are already published? So that's how we came up with the topic on meta-analysis for microarray data sets. Okay, so this PhD was entirely free. We took all the data that we needed from public repositories such as Gene Expression Omnibus, GEO, and Array Express. Okay, and we got a pretty good paper out of it. And the paper is more on how do you do such a study? How do you combine data sets? And there were a number of challenges, but it was very good because using free data, we were able to generate novel insights. Okay, so that's the first. So it was good experience working in a PhD. And then I decided, okay, I want to go and work in a bigger group, right? So that's how we ended up with the my first postdoctoral training, which was in King's College, which was in Imperial College, and we were working on largely on genome-wide association studies, focusing on asthma, allergy, and lung function, right? So, for example, what you can see is on the bottom, the colorful chart is what we call a Manhattan plot, where you're plotting the p-values of the genetic associations, right? And what you can see is there are several areas where there's very highly significant hits, which is the thing I circled in red. Then you can zoom into that region, and then that's what we call locus zoom plot. That means we zoom to that particular locus. We see, yes, it's not a spurious hit. There are other highly correlated SNPs in that region, which are also giving you high significance value. And then you can go back and you can look at the individual studies. So typically for a GWAS study, you need tens of thousands of samples. So depending on the phenotype, but generally you need quite a large number of studies. So then what we did is we went back and we say, okay, let's open up the studies and make sure they're all showing consistent effect. So here, this is a rather small collaboration. It's got 6 studies in it. So we confirmed that the direction of association is the same and the magnitude is about the same for all 6 studies. So what we're seeing is realistic, okay? This GWAS was fantastic. It was the era, we were doing the GWAS era. There's a lot of work. And I think the fascinating thing about this is it was the first time, I believe, a lot of scientists actually openly shared data. Before that, everybody's like, okay, this is my data. I'll work on it. I'll publish. After I publish, I will share the summary results with you. But here, in this time, everybody realized if you have a sample with a GWAS cohort of 2,000, 3,000, it wasn't good enough. So we started collaborating, we started sharing data openly, and we were able to do things that we were not able to do before. So it was excellent experience for training. And for a lot of scientists as well, is how do you share data, right? And there's agreements that you have to write. So there were a lot of paperwork around it. And towards the end of my postdoc training, I started to lead or co-lead 2 or 3 GWAS consortia. And again, that was a different skill set that I had to acquire. Like, how do you manage different times, time zones, participants, and so on. Okay. Then I got a bit disillusioned after about 3 years of doing this GWAS hits, because a lot of GWAS hits are actually located in intronic regions or intergenic regions. So more than 90% of the hits we are finding do not code for a protein, right? 95% of the SNPs. Although it was fantastic and robust science, it still left something lacking. Like, you're trying to understand what is going on, right? So what we do is, then I moved on to my second postdoc, which was in King's College. And this time we were looking at expression QTLs or eQTLs, right? And the main aim is, what you want to see is on the right-hand side, you see, for example, there's a transcription factor gene. You might have a mutation in that gene. And because of the mutation, you may not be able to transcribe the transcription factors. Therefore, you may not be able to target the genes of interest, right? So the question is, and then if you see on the bottom right, you can see AA is the wild type, and then when there's mutations, the expression levels of that particular gene goes down, okay? So we did this in 10 brain regions. We generated the exon level expression as well as the genome-wide SNPs. It was a very highly computational work. We were doing trillions of tests and then trying to get something out of it that we can explain. We got a lot of hits actually, but we found it very challenging to describe this in a paper, right? To compress that amount of information to 3,000 words to build a story, okay? So this was quite a interesting experience, because it's not that we didn't have negative results. We had a lot of positive results, but how do you explain this, right? How do you maximize the value? So one thing we did is we started partnering with a lot of GWAS consortiums. Every time they found a SNP, we would go and look in our database and we say, right, can we explain some of your hits, right? And then eventually got to a point where there were too many requests, so we built a website and we just made it public and say, okay, if you're interested, just go and find it yourself. And the data is also publicly available. So again, you know, it's free to take and work with. Then after another 3 or 4 years, I wanted to go back to Oxford, and I got the opportunity to do so as a junior group leader. So we managed a transcription core facility. What they didn't tell me at the interview is I will be managing stuff in the wet lab as well, and I'll be responsible for processing the sample. I thought it was just the analytical skills they needed, but they needed somebody to manage the wet lab. So that was scary. The first few months was very, very scary, and I actually went into the lab to kind of understand some of the techniques. I still cannot pipe it, but I just to understand the workflow and manage the staff, right? So this was quite an exciting time. So within 3 years, we processed more than 3,000 samples. And I had another junior bioinformatician with me. So me and him, we would analyze the data coming out of this. And then we would actually say, right, this data doesn't look good. Can we go back to the wet lab and figure out what are the steps that was done? Was there any change in the reagent lots? Was the study design wrong? What's going on, right? And we also were able to do a bit of science as well, actually. So this is what we would call systems biology. So that you see the yin and yang chart on the right-hand side. So what we have is very much. So this is on the white circle. You would get the participants in. You would vaccinate them, and then after say 2 to 3 weeks, you would actually bring them back, and you would challenge them. So if it's so I was working in a vaccine institute. So if let's say it's malaria, they would fly in the mosquitoes, right, all the way from Africa, and they would deliberately infect people who were vaccinated, right? And they would take the bloods, and then from the bloods, we would generate transcriptomics data. For certain diseases like Ebola and TB, obviously you can't do the deliberate infection because there's no effective treatment. Malaria, it was okay. Flu, it was okay. So certain things we could. So we had a very good idea of whether the vaccine was efficient or not. But also we were interested in the immune profiles. What are the immune profiles triggered that can indicate whether somebody would be protected or not? Now, with that information, what we would actually go back is we'll go back into the lab and we would say, right, it looks like this particular pathway is really important for immune activation, but it's not there in every human being. So can we add an extra compound that would compensate for this, right? And then Jenner Institute also had the capability to actually do the modeling, the molecule detection, and then they would do it in the small animals, and then they would be able to go back, redesign the vaccine, and go back and test it again in another set of people. So this was really fascinating because in one physical campus, one physical area, we had the vaccine manufacturing facility, we had a clinical trial facility, we had the hospital nearby. Luckily, we never had to use them. We had the labs, we had the sample storage, and then we have the analytical team, and we also had the team that built the vaccine. So all of this was nicely situated and it was very well oiled. So they are also doing something on COVID vaccine now at the moment. So this is fascinating, right? Then as there wasn't enough vitamin D because there was no sunlight. Anyway, I was a bit homesick. So then I said, okay, let me come back to Singapore, which is close to home for me. And what I had, so that brought me back to Singapore, which is where I am. So in Singapore, I take on multiple hats. So one of my staff is tasks is to basically take care of the IT team in GIS. I know it kind of sounds a bit not very exciting, but every day there's a new challenge, and if we don't solve that challenge, the researchers will not work. And what I bring to the whole, I mean, I'm not IT trained, but what I bring to the picture is what are the needs of a scientist, right? Because sometimes the IT folks will just go on about security and all of that stuff. Kashish, maybe I'll take the question later. So Kashish is asking what kind of analysis did I do in the, so what we did is we generated data using microarrays here or RNA sequencing. So if it was animals, we would use RNA sequencing, and we would try to correlate those profiles with, let's say, T cell responses, either through ELISA or antibody responses through ELISpot. So we're trying to correlate those two and trying to find the markers of immunogenicity. Yeah. Very challenging work because there are many things that can affect immunogenicity, including the host response. As Professor Bruno said, the antibodies, they're very challenging. Okay, so we did have some inclination, we did have some ideas about what was causing it. So some of this work is still a bit confidential, so I can't discuss too much about it. But it's fascinating work, actually. Where are we? Yeah. So and also strangely enough, when they built the COVID testing facility in A-Star in response to the pandemic, they asked me to manage the IT infrastructure. Again, there was a lot of learning to do, but I think my bioinformatics training helped me a lot, and I'll explain why in the later slides. My real passion is in trying to teach and train people. So we do a lot of introductory courses, we do classroom style, but we also have a lot of one-on-one teaching. And a lot of this has actually in the last 6 months moved online. And that was another quite interesting aspect. We had to learn how to teach online. So it's not as simple as, okay, sometimes it was simpler, sometimes it was harder to teach online. But that required, I think everybody here had to learn that as well, right? And then when I also got a small group who focuses on the scientific projects. So we focus mostly on bulk and single RNA sequencing technology, mostly 10x and so on. But we also do a little bit on RNA editing and, sorry, that should have been RNA silencing projects, okay? And we work the model as a fee for service. So we either charge our collaborators who can be internal from GIS or can be other RIs. We do charge them a fee for it. We are quite, I think, quite cheap. We're quite generous because we don't just give them the results and say, go away. We actually, even though we charge them for the analytical time, most of it is actually for computing. We use a lot of AWS computing. So a lot of that goes to cloud computing, but we actually will follow our collaborators until the end, until they get a publication, right? So in return, we ask for co-authorship on that. So although I say fee for service, I don't think that we're actually profiting very much. Actually, no profit. But sometimes they don't have the funds. So what we do is we do collaborative projects. So we say, okay, we'll work on you on the first project, we'll help you with it. And then when they apply for grants, they put us there and then a certain amount of funds come to our core facility so I can keep my staff hired, right? So the summary of my career is actually it's highly diverse, both in terms of technology and analytical methods. Disease and biological domains. So I work from anywhere from vaccine studies to pregnancy to metabolic health to editing, you know, so it is very diverse and it's highly diverse who I work with. Sometimes it's the IT folks, sometimes it's the professors, sometimes the students, right? And different domains as well. But what brings them all together is they are all highly computational. Okay, and they're highly collaborative and highly multidisciplinary. So we are in constant communication with our collaborators. We don't work where they send us the data and then 1 year later we just send them the paper or anything like that. So ours is a continuous collaboration. So every couple of months or every week or sometimes depending on the nature of the project, we are in contact with that person. Okay, which is good. So now enough about me. Talked a lot about me. So as you probably know, this might be what you guys familiar with, right? Like the traditional academic pathways, right? Like first you're a student, and then when you're a student, your focus is trying to learn, trying to gain experience, trying to get technical skills, right? And then when you gradually go on to be a master's student and a PhD student, after that, you'll become a postdoc. That's a typical academic pathway. So there your focus changes to more publications, collaborations, trying to get grants, but still primarily I would say publication is what you're assessed on. And then once you're done, you think you're done, then you are like a junior PI or junior professor, and that's when the real stress begins, okay? Because now the expectations are higher. So suddenly, not just publication, but you're going to be reviewed. Your KPI is going to include grants, patents, who you know, how many students have you graduated, are you able to give lectures, and then take on admin work, right? And then later, after a few years, once you have crossed that burden, that boundary, you become a senior PI, right? And now your main duty is admin work, okay? So you can ask your professors, they will tell you how much work is in the admin work, trying to get the paperwork, trying to apply for grants. But now they're not applying grants for themselves. They're applying grants so that they can keep all the rest of us hired, right? And they also play a very important role where they're mentoring junior PIs, postdocs, and they are also responsible for managing collaborations, which basically means they also have to respond to all the funding bodies, right? So this is your traditional pathway. Okay. Any questions? In case you're not aware. Okay. So,
hello, Professor Ramaswamy, sir.
Yep, go ahead.
I'm Professor Krupanidhi.
Hi.
So we are glad to have a few suggestions from you. What type of the what type of the topics that our students can take up for their projects, particularly contemporary interest bioinformatics topics.
Oh, okay. Yep. I will come on to that. So I've not, I deliberately avoided anything scientific because I think there would have been plenty of scientific. I was kind of trying to get more towards the, let me do a couple more slides and then I'll come back and answer your questions. Is that okay?
Okay, thank you. Thank you.
Sure. So the traditional academic work life has a lot of advantages, okay? There's a lot of work-life balance. You know, sometimes I want to work from home, fantastic, I can do that. Sometimes I want to work in the weekends, great, I can do that as well. There's a lot of work-life balance. You have the freedom to pursue what you're really curious. You can always say, nah, I'm not interested in that. I want to work more on this. There's a lot of opportunities to travel, either in terms of presenting at conferences, but also for career-wise. Because one thing I can tell you is bioinformaticians are in high demand because a lot of the, some of the bioinformaticians actually have left to data science. That has created a vacuum both in Singapore and generally in everywhere. Because of the vacuum, there's more demand for bioinformaticians again, right? Especially junior bioinformaticians. And bioinformatics is a really good way to contribute to science and society, okay? But there are certain disadvantages, okay, about academic pathways, because the first thing is, especially in NGS technology, the technology is changing very rapidly. Every 5 to 10 years, there's a major development. So if you think about it, maybe 10, 20 years ago, microarray was hot topic. And then RNA sequencing was the hot topic. And now it's single cell technology, single cell RNA-seq. And then the future is probably going to be spatial transcriptomics, okay? And the other drawback is funding. So both either your own salary or to fund the project that you want. This is why you end up seeing people doing projects with 2 samples or 3 samples because they are very, very expensive, okay? There's also the career progression is sometimes a little bit uncertain. How do you go from a postdoc to a PI? That stage is a very challenging stage. And application for funding is also very difficult because sometimes it's 20 applications to 1 successful grant, you know. And the last point I want to highlight is loneliness. Okay, so this is a real, it's not a fluffy thing because it can be very lonely because you might be 1 or 5 or 10 people in the world who really understand what you're doing, right? And because you don't have that many, once you have reached a certain, when you talk to your professors, right, they will tell you like, oh, I wish more people understood what I was doing, right? When you reach the top, it can be a bit lonely, right? So that is some of the stuff that pros and cons of academic pathway. But if you look at the common skill sets, right, like what are the skill sets that is required for a bioinformatician? The first and foremost is computational skills. Now, bioinformatics itself has many different meanings. It could be protein bioinformatics, it could be next generation sequence bioinformatics, it could be imaging bioinformatics. So there are lots of different bioinformatics, but at the core of it is computational skills, right? So get really good at your computational skills. R, Python, those are the 2 leading programs. So get really good at your skill sets, okay? The other one is data literacy. So how good are you at looking at the data and finding outliers? Can you look at a data and say, look, certain things are rounded to 0.5 and 0.25, but then once in a while I see a 0.367. So what, you know, that, are you able to look at the data and spot those anomalies, right? And part of the data literacy is, it comes through both analysis and coding skills. Then you have mathematics, okay? If you don't like mathematics, please try to learn a little bit because it will take you a long way down. So especially algorithm development and probabilities. So these are very crucial skill set. But there is another aspect which is slightly forgotten, what makes a bioinformatician really good. Communication skills, because you're always talking to biologists, non-technical people, sometimes even among technical people, you know, you still got to explain to them, you got to present your ideas, either in the form of presentation, writing, data visualization. So these are all quite valuable skill sets, right? And where I'm just giving you some examples of where you might want to think about going forward, right? If academic career are not for you, what are the non-traditional pathways, right? So as I said, like managing core facilities, because usually bioinformatics folks are very organized, you know, even from the way you organize your data, your scripts, you're very organized. You're much more organized than most people, right? So managing core facility is not, it does require you to learn a few things, but you can do that. Bioinformatics as a service. I know there are a number of companies in India and elsewhere that actually do bioinformatics as a service. Collaborators. Programming skills, data visualization, teaching, and of course, ultimately, data science as well, right? I mean, this is why a lot of bioinformaticians actually ended up in data science, because they have built years and years of experience working with data, understanding data, and how to communicate data, right? So that's how they ended up with being data science. So don't just look to universities and research institutes, but look into hospitals, pharma companies, software companies, even journals. A friend of mine was recently just hired into a patent search company. So they're actually mining patent data to look for which patents are worth funding or purchasing further. So they have taken up quite a number of things. I have a few more slides, but I want to answer the question, what kind of topics and projects can your students take on, right?
Yeah, please, sir.
Yeah, so it's a bit challenging because a lot of this depends on data availability, right?
Yeah.
Yeah. So either you can go down the route of analytics. So that means you partner with somebody who has the data. They give you the data set, you analyze for them, and you try to find novel insights. Okay, that's one route. So for this, you really need to have strong collaborations with people who have the data, right? And the other option you have is you can, if you want to go down, let's say the algorithm route, you can do what I did, which is basically try to find publicly available data sets and try to mine for valuable insights. Now, I know I kind of say it's a free way of doing a PhD, right? But actually it gave me a lot of useful things because you're reusing data and you actually, the results you find in one data set, if you cannot reproduce in another data set, then that result is not valid, right? So you're actually building robustness into your data. There are a couple of websites you can look into for single cell. So single cell is a very hot topic at the moment because the cost of doing single cell has come down to a more affordable price. But you can also do bulk RNA-seq. So these are the two things that I would recommend. What are the backgrounds of your students? Are they mostly master's, PhD students?
Our students are with the BTech Bioinformatics, sir, with a lot of inputs on information technology tools like Python, Java, and all those things.
Perfect. Yeah. So Python is great. I would recommend looking into R as well because R has a lot of what do you call biological packages built in. So Bioconductor, for example, is a website you can go to. It ultimately you have to ask the question why, right? So if you want, is it just, if it's for algorithm development, no problem. There are lots of data you can download, try your method and find that.
Algorithm development, is it computational, what is it called, languages or programs need to be used, or already existing softwares need to be adapted, sir?
No, you have to come up with the algorithm, right? So like the single cell RNA-seq, one of the challenges is for cell identity. So you can do as a fancy, like it's a little bit like what Bruno also described earlier, right? So that's also algorithm development, right? So you got to understand the problem well enough. And then you got to say, can I tackle it in a different way?
Yes, sir.
Right?
Yes, sir.
Either using machine learning or AI techniques and so on.
Yes, sir.
Yeah.
Thanks. Thank you. We'll be in contact with you.
No problem. I mean, we can brainstorm later.
Yes.
So I don't know, I feel like we're all here talking about some of the important things I learned, right? Like, I think it starts with saying I'm worthy to contribute, right? I'm worthy to speak my thoughts, to listen to others, to make a change, to teach others, right? So you got to build, for students, I'm talking to students here, right? Like you got to build your presence. So I find a lot of students tend to be very shy, very quiet, and then they say, oh, I'm not good enough. So that's usually the first challenge I have with the students is to say, nope, you're good enough, go and do this, right? So for the students, this is the first thing, right? The second thing is you can think big, but you need to do some action now. Don't say, I want to go solve cancer. Great, that's fantastic, but how are you going to do that? Break it into actionable plans. Each plan should have a aim and a goal. So that means when you have reached that goal, you can, you know that you have achieved that. So again, use your mentors, lecturers, teachers who are there to help to help you plan these. You have to make it into manageable chunks. Okay. And who's the best person to lead? Is it you? Is it somebody else? Right. You want to solve something ambitious, great. But if you don't have the skill sets to do that by yourself, then go and find somebody you can contribute to, right? You gotta share, and don't be afraid to fail. And especially when you fail, share the failure, right? Because if you don't share, somebody else will fail doing exactly what you did. So you want to help others as well. You want to do something worthy, stay current. You want to find out what are the topics of interest. I think this is the question you asked me. Talk to people in the field and think about implementation, right? How are you going to come out? So the last statement there, how do you get up in the morning, right? This is my question. Like if I get up in the morning and I'm excited to go work, that means I got something interesting to do. If I get up in the morning and I go, you know what? I wish I'm going to call in sick. That means I'm not doing something useful. That means I need to go and take some action. Okay. So that's why I say, how do you get up in the morning? Know your purpose, right? Why are you doing bioinformatics? Or why is it to win something? Are you to win some prestige, to win accolades, to win acknowledgement, which is fine. We do need people who are competitive and who want to win. Or are you doing it purely out of curiosity because you want to discover new things? For me, it's because I want to help other people do better research, right? So when I was playing the PI role, in the PI role, I was expected to be more competitive, to go get grants, to win, compete people, publish before others do, which, I didn't do very well, but that wasn't very satisfactory because I was in the wrong role. So when I moved to a more support role, I'm way happier. Okay, my salary is exactly the same, but I'm way, way happier than being in the PI role to compete. Okay, nothing wrong with either roles. All roles are great. No, I don't work with plant-based data. Sorry. Okay. I think the last slide I have is two slides. Communication is key. So you need to be listening as well as speaking, right? It took me many years to actually start listening to people because I thought communication means you just talk, right? And sometimes people are complaining to you or they're saying something, but you got to understand why they're doing that, right? So that's another skill set to have. The third point is a bit controversial. Manage your boss, okay? So if you come from a very hierarchical society, you might be scared to tell your boss off that they're in the wrong direction or that they're giving you too much or too little. But you got to understand your boss. Your boss has a boss, and his boss has a boss. So the instruction is flowing down, and what are they trying to deliver? And work with them. And being a good follower is just as important as being a good boss, right? So that's what I mean by manage your boss. So in the sense that understand your boss, see what their priorities are, and try to explain what your priorities are, okay? It's not an easy skill set, but it will take time. And then the last slide I have is that the teamwork is crucial. So I think there's this famous saying, right? If you want to go fast, go alone. If you want to go far, go in teams. So I don't know who actually said that. But my first postdoc boss, Debbie Jarvis, who's amazing. She's got like lots of collaborations. And I was a very hot-headed postdoc. I was kind of like always telling people like, oh, you're wrong. You should be using this program. This is not right. And she sat me down and said, look, if you want to work on your own, that's fine. You're going to be 100% right or 100% wrong. But when you work in a team, you got to be prepared to be a little bit wrong, right? So, and that's where it really opened up my eyes. And he said, okay. So it's not about being right all the time. It's about working with people, trying to find that common ground, right? So this is probably the best advice I got, and I'm trying to share that with you all. Okay, and I think that's it. Yeah, that's the last slide I have from here. Maybe contact me later about like potential projects and stuff. I can try to maybe have a chat later. Okay, thank you. Any questions so far, or?
Thank you very much, sir. That was very inquisitive. Now we are open for a quick question and answer session. Would anyone like to raise a question?
Please ask anyone for that. Okay, then we will move on next speaker.
All right. Thank you very much. I thank all the delegates who have shared their views and delivered lectures. I conclude with this. This is the end of technical session 1. Now I would like to introduce Ms. Suhana to take over the technical session 2. Ms. Suhana, please carry on.
Good afternoon. Now I'd like to take the privilege to present before you the profile of our next speaker, Dr. M.V. Keat. Dr. M.V. Keat is an associate professor at the School of Data Sciences in Perdana University, Malaysia. She is currently acting the dean and head of the institution education unit at School of Data Sciences, Perdana University. She obtained her Bachelor of Sciences in Bioinformatics from the University of Malaysia in 2004. Before obtaining PhD in Structural Bioinformatics from University of Science, Malaysia in 2010. Dr. Yam started her journey in bioinformatics since undergraduate years and went on to specialize in structural bioinformatics during her PhD. Years lately, she has started venturing into education, research, and innovation with a special interest of reaching out the community about bioinformatics and data sciences. Over the years, she has obtained several research grants from the Malaysian government and university to conduct bioinformatics related works. She nurtures postgraduates and undergraduate students, research and interns throughout her career as an academic staff. Besides that, as a certified trainer herself, also actively trains and conducts trainings, workshops across Malaysia and Southeast Asian topics in bioinformatics, data science, and innovation and education. She is currently the Vice President, Education and Research for AP Bionet, Asia Pacific Bioinformatics Network. She is also the founding chairperson of My Bionet InfoNet, a virtual networking platform for Malaysian bioinformatics enthusiasts. She also serves as an EXO member for the Malaysian Society of Bioinformatics and Computational Biology. She was an affiliate member of the Young Scientist Network Academy of Malaysia and was a member of Royal Chemistry Society. Now I'd like to invite Dr. M to deliver her talk on structural bioinformatics in drug discovery.
Thank you very
Ma'am,
much for
please.
the introduction. Just checking if you can hear me.
Yes, ma'am, you are audible.
Great. I'm
Please
going to
go
share
ahead.
my
Yeah.
I'm going to share my screen now. So please also let me know if you can see my screen.
Yes, ma'am, it is.
Great. Thank you very much. So good afternoon, everybody, and thank you very much for this invitation. It's certainly my honor to be here. So as introduced by both the chairperson, you can see that I'm actually from Perdana University School of Data Sciences, and my topic today, I'll be having quite a light topic because I thought maybe around this afternoon time you'll probably be prefer to have a light topic. So I actually decided to talk about structural bioinformatics in drug discovery, but I'm not going to talk more about the sciences, but more into whether is it still relevant today in today's context. Yep. So please allow me to share a little bit with you about where I'm from. I'm currently from Perdana University. As you see, this is a Malaysian map. So if we can zoom in, we are actually in Kuala Lumpur, very much downtown at Kuala Lumpur, the city center of Malaysia. So our building, our office building is currently at Damansara Heights, a very upbeat place in Kuala Lumpur, where we are connected with a lot of public transportation and easy access to many famous places in Kuala Lumpur. So this is how our campus look like. We are a one building block kind of campus with focus with our students. And just to share some significant milestone for the university actually have two medical schools. Uh, Two medical schools that we started in 2011. So we are relatively a new university. And at the same time, we also set up another school of foundation where we cater for pre-university students. And moving on, our school of data sciences actually was set up in 2015, and only last year we set up school of business. So you can see that the university is basically quite new, and we are still in the midst of expanding. Yeah. So I'm going to move into talking about structural bioinformatics. I guess there are a number of people here who are probably interested in structural bioinformatics. But for the benefit of everyone, I'll probably just give you a very brief introduction about structural bioinformatics. This is basically a subdiscipline of bioinformatics whereby the main emphasis of this field is to focus on the representation, storage, retrieval, analysis, and display of structural information at atomic and also subcellular level. So whatever that we are looking at is actually very much into the atomic level. And of course, you can see that we have two main goals when we perform structural bioinformatics. The first is to create a general purpose to manipulate information about biological macromolecules. So we are basically focusing on the macromolecule. And another thing is to, of course, to apply enable to solve any problem in biology, and also ultimately is to create new knowledge. But there's always a very strong belief that in order to perform effective structural bioinformatics, there must be an availability of high resolution structural information about the biological system. And why do we need that? Because only high resolution information, high precision of information will enable us to get very good reasoning into the function and also the effects. So if we wanted to understand about a particular protein, about their function, about their how they perform their role in the particular system, it's very important for us to get very high quality input data. Next, I'm going to move into this perspective of looking into structure, because when we perform studies on protein, or even perform studies in DNA or even RNA, we might encounter into either structural perspective or even sequential perspective. And with today's current scenario, we can see that there are actually a lot of people involved in NGS, which is very much into the sequence. But the importance of structure, as specifically in protein, is that a structure is actually 3 to 10 times more conserved than sequence. And because of that, that is so important that in order to get a better understanding of their responses in the biological process, it's very important for us to understand the structure and also to get hold of the structure. So these are some of the common input files that we see from a bioinformatics. I would assume that most of the participants here are bioinformatics background, or perhaps you perhaps are from biology background, but you wanted to get into bioinformatics because of various reasons. So these are some of the common input files or even output files that we see in order to let us perform our job. But what I wanted to bring you in is that a lot of the time, even as a biologist yourself, that when we talk about protein, this is perhaps something that you imagine, yeah, because protein for a lot of people, especially those working in the lab, is always in the form of powder. But for a lot of bioinformaticists or even researcher, we often see this instead of this. And as a structural person, I would personally look into this as well. Yeah, so if you ask me how useful it is to look at it this way or this way, I would say obviously this way. This is a three-dimensional way that allow us to have so good understanding about the particular structure that we're working on. And of course, as technology progress, we are also looking into this perspective. This is a 3D printing of a dengue virus. That is actually my hand, so you're seeing that. So that is actually the size of a dengue virus capsid. And of course, with the advancement, now we are moving into augmented reality and also virtual reality in understanding structural bioinformatics. So moving on, structural bioinformatics is no longer just a 1D or 2D thing, just using your eyes and view it either on your screen, but we're looking into a more personalized feeling whereby you can touch, you can feel, and perhaps you can perform even some of the work with your hand as well. So this is where the field has evolved from, I would say, about 20 years back until today. Things have changed tremendously. So I also wanted to talk about drug research. I mean, this is probably one of the reasons why we have this gathering today. We talk a lot about drug. So a lot of time, this is represented, I guess if you ask a lot of people who are in drug discovery work, they would tell you drug research is like the search for a needle in a haystack. It's totally fun, I would say, fun and challenging. So just to find that little tiny needle in that haystack could probably take you a very, very long time, and of course with a lot of money that is involved. So this is actually the process that you can see. For a traditional drug discovery way, it actually begins with just doing some target identification, target validation, lead identification, candidate optimization. Now, this entire process itself could take you up to about 10 years before you can even bring something quite promising or something quite solid into preclinical. Now, preclinical is the part whereby you perform some, say, like animal studies or even perform it at a cellular level. So this is what we call preclinical. And of course, finally into the clinical stage whereby we are actually experimenting with real human subjects. And this could take up to many years just to check on the effects. So this is in terms of the time and in terms of this is in terms of the time. But if you look at in terms of the compound, how much compound we actually get involved. So this is actually again a very simplified model to show you that, if you start off stage 1 doing drug discovery with only 10,000 compound, when we move to stage 2, ultimately we are only interested to look into about 250 compounds. I guess the reason is very simple. If you are doing it in the wet lab, you can't be bringing all 10,000 compound into the lab because we are looking into the cost, we are looking into the time. It's simply too much to waste, or if we can't be assured of the success rate, perhaps there should be a way for us to do this filtering process. And by the time it reach into the clinical stage, it should only be about 5 compounds. And these 5 compounds also eventually goes through the phase 0, phase 1, phase 2, phase 3, and ultimately phase 4. So even at the clinical development itself, there are already 5 phases that scientists have to go through. And of course, finally, that particular one compound that managed to make through into the market, whereby it has to get the approval from the regulatory board, FDA would be the common ones that we see. Well, even each and every country has their own regulatory board. For each and every drug to get approval, that actually takes a long time. So to sum up this whole thing, we could see that to develop one drug, to even make it into the market or even to put it into our mouth for consumption, we need to spend approximately 1 billion US dollar and take approximately 10 to 15 years. Now, this is a very, very long process, especially in a desired situation like what we are currently facing now at COVID-19. We certainly do not have 10 years to wait for COVID-19. And therefore, we a lot of people has been discussing whether this entire stage can be shortened, can be made into a very fast way so that, we can get at least some candidate to work on, especially now during this COVID-19 situation. All right, so to address that, in drug discovery, this is a traditional way of doing drug discovery, whereby I look into a more computational perspective because we are in bioinformatics. So this is very much into the work. Pardon me. Okay, yeah. Computational work. So you can see that in terms of the drug discovery, there's actually two major field, the structure-based and also the ligand-based. So for structure-based, we often heard about ligand, docking, homology modeling, quantum mechanics calculation, molecular dynamics simulation. I believe these are some of the methods that you probably have heard, but maybe you're not very sure what it is. Please allow me to explain it in a while, in a very brief way. Hopefully, you can get it understood. And of course, besides the structure-based, we also have ligand-based. So you probably heard of QSAR, pharmacophore, or even similarity searching. And finally, we could combine them. Hopefully, we can better optimize the lead. And finally, it can be a better drug candidate. So to sum up the whole thing, again, in today's context, we are not only looking at the traditional drug discovery work, because we need to be a little smarter, I would say, or more rational. So there are a lot of combination kind of methods that could be used in order to make this method to be effective, or hopefully to shorten the entire duration to discover a potential inhibitor or even drug. So like in today's context, we talk about NGS. There are a lot of human genome sequencing that's being done. So a lot of NGS work being done. And of course, from a proteomic person and also from a structural person, I will always ask my colleague and say, do you have anything interesting from the NGS that I can take and continue with doing drug discovery and all? So these are all very important questions that we need to ask our counterpart. Yeah, and also the development in terms of three-dimensional. So structural perspective, this is also about getting the data. How do we get effective and also good data? And of course, for most of us who are in structural bioinformatics, we are basically at this level. So a lot of work are actually being done at doing the database of active compounds, or even target and also binding site identification, or perhaps doing some docking, or also some virtual screen, or maybe some molecular dynamic simulation, and of course, finally get it tested in the lab. All right, so I talked very lengthy about structural bioinformatics and also drug discovery. So how do we connect them? Yeah, so we often use structural bioinformatics as a tool to help us to solve various problems. So in terms of in silico protein structure prediction, there are three main methods that it could be used to do structure prediction: homology modeling, fold recognition, ab initio, and of course, performing molecular docking studies, such as something like this. So if you can perform molecular docking simulation in an iterative way, and if you can perform it over 10,000, 100,000, or even million times, we actually name it virtual screening. So this kind of work of doing virtual screen actually allow us to understand or even study whether any of these chemical compound can be used to bind on a particular drug target, and whether it is useful for us to move on to test them in the lab. And of course, if you can see, there's also a different kind of structural work that can be done, because it's not just about getting it docked at a particular target site, drug target binding site, but we also wanted to know what happened after you bind them if we put them into a simulation and try to run it for a period of time, and what will happen then, whether the ligand will continue to stay at the binding site, for it to have any informative or even what is the word, any nice activity, or nothing happen. So it's very important as well to move into the simulation perspective to check how well they actually stay. So I will share with you a little bit of some of the work that me and also my students have performed in the past. So we actually focus, this work was done about 9 years back, that we actually focus on plant. So this plant, Ganoderma lucidum, was used to study on the potential of anti-HIV, and we had quite some nice result to move on. So we actually performed this in computational perspective. And a few years later, we did something similar, not on Ganoderma lucidium, but on understanding toxoplasmosis, which is again a very, I would say, an NCD, and a lot of this kind of disease that has been neglected by a lot of us. So we wanted to study, in terms of computational, whether we can have meaningful meaningful insight on the particular drug target. And back then there was no drug as well. So for us, the objective was very clear, was to try to determine whether there's any potential inhibitor that we could develop from computational method. So few years back, I also work on this work, which is on simulation, where we study on antibiotic. So understanding antibiotic again is very important, because that actually changes how antibiotics should be taken. So this was a work done by me and also my team back then, that we were studying, and we simulated it using molecular dynamics simulation. And besides this work, we also did some calculation just to understand like how much all of these antibiotic actually stay in each and every of the target, and how are the effects like, in order to keep them in the binding site. Of course, the ultimate aim of doing this kind of structural bioinformatics work is actually to understand, and it's actually to develop drug as well. So this is a very good summary that I pull from this paper that talks about what kind of drug that has been successfully developed using computational way. So you can see some of these drugs that's already in the market, and these are the drug target, and what are the disease that they actually target, and what are the ways they actually use. And we are talking about computational ways. So this is solely some of it is structural bioinformatics work. Some of it is more into ligand based. Although it's not really structural bioinformatics, but because it utilizes computational way, so somehow I think it's also wise to mention here as well. So the success rate of doing computational work, and from computational all the way to getting the final drug, is actually there. So it's not saying that that there is none. There are a number of examples which you can see now, and that shows that how important it is to use bioinformatics work to actually complement into doing this kind of drug discovery, and ultimately being a very successful drug that is being targeted into the market. So next, I also wanted to talk about how the big data that we are currently facing now, and how do we relate them with bioinformatics. So this was an article published by Caltech on about big data. How big data has actually influenced computational biology, or even structural biology, in shaping the future of our health. And also there are many other different perspectives that we can look at. So you can see this was a paper published in many years back that talk about the challenges of big data. And although big data is something that we talk about, very frequently probably for the last 3-4 years, but actually big data in biology already happened about 9 years back. That was because back then there was already a data explosion. Well, this kind of data explosion actually happens because of genetic sequencing. So this was quite an old data, I would say. This was in 2012, that was 8 years back. So you can see in terms of the data pile, the data pile is actually just 1/10 the size of the data storage at CERN. So if you know about CERN, CERN is actually the Europe Particle Physics Laboratory near to Geneva, Switzerland. So this data pile is actually only 1/10. So you imagine every year in CERN, they actually produce about 15 petabytes of data, and that is also equivalent to like what we are seeing currently in the biology perspective. So the European Bioinformatics Institute currently stores about 20 petabytes of data about genes, proteins, and so small molecules. And this genomic data is about 2 petabytes per year. All right, so you can see from this perspective, is projected in 2025 that genomic data is going to exceed even whatever data that we see, Twitter, YouTube, or even astronomy data. And just to give you a context, how much is petabytes? You can see from this how many zeros here. So a data scale here will help you to imagine what kind of data that we are talking about. So we are talking about 10 to the power of 15 kind of bytes of data that we see on a yearly basis. And there's also a dramatic decrease in terms of time and also cost to sequence data. So you can see that in order to sequence data, it was 100 million in 2001. And in 2016, it was only $1,000. And today, it actually drops to about 10. So you imagine the cost to actually perform this kind of genomic information is actually so small nowadays. And this is, again, the same information that we are looking at. So if you can see again, in terms of real time, I actually took this data on 15 December, about 5 days ago, from GenBank. You can see how much data that we are actually producing. And using this data, we can actually see that, there's so much potential that we can do with this data. And mind you that this data is actually available free of charge. You don't even need to pay a single cent to get this data, and you can just get it download, to your own to your own PC. So again, perspective wise, this is on the GenBank. But if we look at the structural data as of yesterday, it was only 172,000 structures. And you can see this number is actually very, very small compared to the gene data that you get from the GenBank. And on top of this complexity of not much data that we get from 3D structure, the good quality structure, because data has to be good quality. And just when I started my talk, I actually talk about how important it is to get good quality data. And you can see that good quality data that falls into this region is only this, and it is very, very small. So these are usually some of the I would say the usable data that we can do in order to perform the drug discovery work. Yeah. So this number is actually so small that even we felt that although we are in structural bioinformatics, but because of the complexity of structural information and not able to get a good data source, so there are different ways. There's definitely a need to improve on this particular way. So if you can see that, AI has come into the picture of structural bioinformatics work, where you can see artificial intelligence or AI. Basically, the definition of AI is to mimic human behavior by simulating human intelligence using computer methods. So if I sum it out in AI, there's actually 4 main approaches that we can see from AI perspective. From the machine learning perspective, the deep learning, natural language processing, and also finally robotics and IoT. Although 4 of them sounded the same, but actually they are totally different in AI, where you can see machine learning very much focused on the input data that we get, and we repeatedly optimize it using the data we generate. That's called machine learning. While deep learning is very much into getting the knowledge out of it by simulating the neural network. So this is a deeper kind of learning curve for that particular system that you have. And of course, some of us are also involved in NLP, natural language processing, whereby this is more into automated speech recognition. Yeah, and also we look into robotic and also IoT. So this is more into collecting the data that we need from the right hardware that we required. So this is into the robotics and also the IoT. So how do we get all this AI into drug discovery? So we have heard about drug discovery, heard about AI. Now let's try to combine them together. Although it sounded a bit challenging, like how we can do that, but actually, there are a lot of ways and a lot of effort in putting AI into drug discovery. So if you remember, I vividly explained about this just now when I started my talk. And let's see how we can actually combine machine learning into this pipeline. So there are a number of generative model that we can fit into machine learning, including reinforcement kind of learning at this level, at the screening level. And once we pass the screening level, when we go into the filtering level, this is where we can put into deep learning method. And finally, we can do some graph embedding to make it to make the system learn better. So not only combining this, but you can actually see that there are actually a number of studies being done, like how much, what is the need of combining AI into drug discovery. And you can see that 36% of global AI in drug discovery market was actually done by deep learning. So this is actually to show the success rate of doing that. And if we want to see what kind of who are the main stakeholders in putting this AI together with drug discovery? Of course, can't forget, pharmaceutical companies. They are the major stakeholders into doing this. And of course, we have there are also AI introduced into it. And of course, there are some technology company as well. So you can see these are all the three main field that comes together in order to bring AI into healthcare and also drug discovery. And this is another perspective of how where are they involved specifically at each and every stage. So even started from the target discovery itself. So you must bear in mind that for some of us that is doing structural bioinformatics, we are very much involved at this level, the preclinical level. So preclinical level, of course, is the part where we use a lot of AI to help us to speed up our entire discovery process. And if you can see this as well, these are some of the example of the AI companies that is being active currently in doing drug life cycle as well. So these are the big companies, how much they have invested into producing a good drug so that it can get into the market. And these are just some of the snippets that I managed to pull through from some very important literature about AI in drug discovery. So there's one here, another one here. Of course, they're studying at different perspective as well. Not only studying at some of them study at drug discovery, but some actually study even at the biologist system itself. So if we conclude all of these things, you can see that what are the challenges that we have. So the first challenge that I can see is that it's a data challenge. Why do I say data challenge? Because there's obviously a lack of high resolution 3D data. Just as how I've explained, the data that we have received from PDB is probably only about 10% of it compared to the genomic data. And I would say and out of those data, only about 10% of it is really functional. Now, what do I mean by that? I'm talking about in terms of the quality of the data. So it's also, again, important to look into the resolution of the data and how much we can trust the data. Second challenge, I will probably put into technological perspective. There's obviously a need to improve techniques and also method. Mind you that some of these method actually is as old as probably 50 years old. And I'm not saying that they are not good, but it's obviously need to have a little tweak here and there to improve. And perhaps with the AI method that I've just introduced to you, perhaps it's actually time that, we start to combine AI method with some traditional knowledge base, and maybe we can enhance that with a new algorithm and hopefully the entire simulation or the screening process will be done even in a more accurate way. And another important problem that we're actually facing is actually storage. Today we're talking about petabyte, and let me ask you, how many of our lab actually equipped with this hardware and also the cost to help us to store this data? So this is also another challenge that we face. And most importantly, I would say in terms of the validation. So it's not just about doing this in the computer lab, validating against with the database, but there's also a need to validate with experiment and also clinical studies. And obviously, we need to work together with other fields. We can't do this all by ourselves. There's always a need to do collaborative work with other labs and also other fields. So if I were to sum up everything, there's this 5 main key consideration when we wanted to adapt this AI solution. First, it has to improve the access to robust, reliable data. Secondly, it must be able to recognize the disruptive potential of tech giants. Third is to increase the diversification of drug discovery pipeline. We're not just looking into the traditional pipeline, but we are looking into a more creative way. And of course, acquiring skill. And when we talk about acquiring skill, we actually talk about the scientist, the researcher, are researchers actually ready to do this? And of course, finally, the critical thinking part, and also the performance indicator. So I actually echo with what Dr. Adais has shared before this. Doing bioinformatics is not just about skills, soft skill. Sorry, it's not just about hard skills where you move into the programming part, the analytics part, but it's also a lot about that mind, that critical mind. And I'm going to say that a lot of us are probably not trained, to think critically. That is why it's so important to have a team to brainstorm together. So before I end my talk, I just want to bring up this topic again, this question. In fact, this is the title of my talk. So my question is very simple. In today's context, is structural bioinformatics still relevant? Now, my simple answer is yes, it's a yes. It's not just a yes, but also improve. So there is importance. It's so important to improve on the method that we have, not only doing the traditional structural bioinformatics way where we used to or we are comfortable in doing, but it's perhaps time that we have to look into using AI or even help from different kind of technology to help in that perspective. So this is actually my contact information. If you're interested to find out about our team, our group in Perdana University, we do have a bioinformatics team in Perdana University, which we not only work in structure, but we also touch on various perspectives of bioinformatics. So you can get in touch with us. Please like our Facebook, our Instagram, our YouTube channel as well. And if you have any question, feel free to ask here. So with that, I'm going to end my talk here. Thank you very much for your attention.
Madam, yeah.
Yes.
I'm Professor Krupanidhi.
Hi, Professor.
Yeah, we wish to know what are the AIs, because we are basically with the biology background, biotechnology background. You have been stressing much on AI skills.
Sure.
So may I know a few?
Okay, so if I could,
which are useful in structural bioinformatics.
Yeah, so if I could explain to you, Professor, we should always start with the basic, which is the structural bioinformatics. So there are a lot of perspectives that we need to understand the structure-wise, the protein structure, and also what are the methods that we can use. So probably some of the methods you have heard, like docking simulation, ligand-based drug discovery, like QSAR, molecular dynamic simulation. If we start with all these methods, and then whatever method that we can use to improve on that, those would be an additional on the artificial intelligence method. So Professor, maybe you can start with the structural work first. Then on top of that, we can use all the artificial intelligence method to help us to I would say, fine tune it to make it more presentable, and hope that we can filter the result into a better way. I hope that I answered your question.
Yeah, that's true, madam. So is there any possibility for our students to come out to you for internship, madam?
Oh, sure, definitely. Something that I forgot to share is that in Perdana University, we actually accept interns. So we accept a lot of intern students into our university to do their internship. But because of the current lockdown, most of them couldn't come into the country, but we actually accept virtual internship. And the school also perform a lot of training, not only on structural perspective, but also in bioinformatics training. So Professor, if you're interested, you can get in touch with me or even go to our Perdana University website to have a look. What I can do is that I can drop my email address in the chat box. So if you have any question, perhaps we can communicate from there. Thank you.
Thank you very much, ma'am. It was really an insightful lecture.
Thank you.
After such an emphasis on AI and structural bioinformatics, we really feel honored to have you, have your esteemed presence amongst us. Now, moving ahead, I would like to take pleasure to present the next speaker, Dr. S. Anil Kumar, Department of Biotechnology, Vignan's Foundation for Science, Technology and Research, for the next talk on the known unknowns of COVID. The known and unknowns of COVID. Sir?
Dr. S. Anil Kumar has obtained his master's and doctor's degree from Osmania University. He has completed sub-national postdoctoral fellowship as a principal investigator from JNTU Hyderabad. Dr. Kumar has 2 years of teaching experience as assistant professor. He is an editorial board member of International Journal of Clinical and Biological Sciences. He is a DBT expert committee of IBSC. He worked as a research associate at A-Star Singapore for developing plant stress protein database. He has 16 publications in various peer-reviewed journals with a cumulative impact factor 50.9, H-index 9, I-10 index 9, and 282 citations. He has 10 book chapters to his credit. Dr. Kumar has received 2 gold medals and 2 Young Scientist Awards, Biocluse Innovation Research Award, AP Bionet Travel Grant Fellowship submitted vector to adjunct, and his image got accepted in BMC Image Library. He is well versed with plant molecular biology, tissue culture, and bioinformatics technique. Now I'd like to invite Dr. Anil to deliver his talk on the known unknowns of COVID-19. Sir, please.
Yeah, thank you, Suhana, and thank you, Akila, for introducing me. Now I'll present myself, Dr. Anil Kumar, working as assistant professor in the Department of Biotechnology, PFSTR University. And my topic for today's presentation is the known unknowns of COVID. We all know that we are battling with coronavirus, and after in the history of humans, after bubonic plague, this is the worst situation which we are facing. And several symptoms are associated with this particular thing. And this coronavirus, it belongs to a group which causes simple common cold to fatal diseases like common cold, MERS, SARS. And this coronavirus recently, it was, a year ago, it was discovered in Wuhan. And it shows several symptoms like fever, mucus production, dry cough. Of all these things, shortness of breath is one of the most important things. If one is feeling shortness of breath, he needs to consult a doctor immediately. And the size of the droplets may range from maybe more than from 5 to 10 microns. And with the help of high technology like high nebulizer, they are identified that they are able to survive in the air for a time period of nearly 3 to 16 hours. And the most important thing is, there are various aspects about this COVID-19. Even if a mother is infected, she can feed her baby. And there is no transmission of coronavirus from mother to a baby when she is carrying. Now comes the importance of this bioinformatics. With the help of bioinformatics, now we are able to develop a vaccine almost within a period of 9 to 10 months, which is very short span for developing any vaccines. In general, if we try to develop any vaccine, it will take years. But during the COVID time, the researchers across the world, they are sweating their blood to find out the solution. And luckily, we got few vaccines, and some countries even started vaccinating their people. And with the help of the bioinformatics, we came to know that after doing phylogenetic analysis of more than 160 SARS-CoV genomes, they had identified that the SARS-CoV is mainly from beta coronavirus and the transmission is mainly from bats. We know that coronavirus has devastated the life in all the fields, mainly economy, agriculture, tourism, energy, health, sports, automobiles, IT, and communications. Especially in the field of healthcare, except the medicine that are related to this, that it has devastated all other medical fields. And this is the non-exhaustive list of medicines or the compounds that are under study for COVID-19 treatment. We can see different compounds or agents like lopinavir, hydroxychloroquine, ivermectin, flavipinavir, rhodoptil, remdesivir, based on their ability to bind or inhibit or prevent the subsequent replication of that particular virus. We had several compounds are in study, and of these things, remdesivir and flavipinavir are most widely used because they want to be very potent in controlling this particular COVID-19. And we all know that a vaccine has to come, it has to pass all these phases starting from pharmaceutical to the last phase, studies after authorization. It has to pass through clinical, non-clinical, all the things. During this COVID, the scientists have developed alternative, they're easily identified the compound which can be developed into vaccine so that it can be easily developed. Because with the help of the bioinformatics and especially for the therapeutic diagnostics, they had developed this vaccine within no time, within the span of 10 to 11 months only. And the key activities for any vaccine adoption should be, it should be available and it should be administrable. It should not have any side effects like allergens or something related to neurological disorders. And it should be readily accessible so that it can be available for a large number of people. acceptable means if it is fine, public will accept that one, and it should be affordable. Why I'm saying this should be affordable means, well, the Pfizer vaccine, it's costing around close to $17. On the other hand, this vaccine that has been developed by the Moderna vaccine is close to $39. So all the developed nations have started this vaccination, and because they have good facilities of research and the transportation, distribution, everything, so they have started this vaccination. But what about the fate of the developing countries or the poor countries? And there was an organization called Gavi, Global for Vaccination and Immunization, okay, wherein it's raising funds for the for the application of the vaccines, or for giving vaccines to the people who are residing in poorest countries, and especially this is focused on giving vaccines to children. Yeah, we can see the list of the approved vaccines by different countries. Switzerland, Malaysia, Britain, Bahrain, Canada, Mexico, they had approved the use of Pfizer vaccine. And in United States, they had approved the use of Moderna vaccine. And in Russia, it was prepared by Gamaleya Institute in the name of Sputnik. Now they are using that one. And India is ready to go for the vaccine, maybe from Covaxin developed by Bharat Biotech or some other things. What is the fate of this? What is the exact update of the COVID-19? Now we already entered into 51st week, and everyone is rolling out their arms to get vaccinated. And there are some concerns associated with these things. And there are also some myths related to these vaccines. Some are saying that if we take the vaccines, they develop some unwanted results like bed in females, and even it reduces the sperm count in males. There are several things associated with this. Most of them are myths, but very rarely they are proved. So it's better to go for the vaccination at the moment so that we can save ourselves. And above all, the social distancing is the best thing so that we can easily get rid of this one. And myself and my team, including my professor and several other colleagues, we had identified potential xanthons which can be used as antiviral compounds, especially targeting this COVID-19, wherein we had identified brachyxanthone, chamixanthone, chlamydia A, B, and different types of those xanthone compounds. We identified those compounds and docked with 6 proteins of this COVID, and wherein we identified that 3 of them are very potential in doing this. Later, we did the molecular dynamics molecular dynamics and simulation studies, and we had identified 6 of them. So which we had communicated to Frontiers in Molecular Science. So I couldn't reveal all those details at the moment. And I'm very thankful to the organizers or the InSyB team for giving me this opportunity, as it was already a statement said by Isaac Newton. Always a scientist, I will stand on the shoulders of the previous one to see the further thing. And it was proved in terms of COVID also again, wherein we have developed taking all the data that is available in the public domain database of the private things, and we are now ready with the vaccines. Thank you one and all.
Thank you very much, sir. That was enlightening. Now it's time for Inquisitive Minds. We are now open for question and answer session. All right. Assuming that no one is having any question, moving forward, now I would like to take immense pleasure to introduce the next speaker, Mr. Patricio Gonzalez Pone, Faculty of Agricultural and Forestry Sciences, Universidad Maule, Chile. Suhana, please take over the session.
Thank you.
Mr. Patricio Gonzalez Pone is a 5th year biotechnology student studying at School of Biotechnology and Engineering of Faculty of Agriculture and Forestry Sciences in Catholic University of Maule, Chile. He is working in his final year thesis under the supervision of Dr. Aparna Banerjee in the area of hot spring associated bacteria and its biofilm production. Now I would like to invite Mr. Patricio to talk on the topic computational study on temperature-driven structure function relationship of biofilm producing glycosyl transferase enzymes. Sir, please.
Okay, thank you. Is visible presentation?
Yes, it is.
Thank you. Okay. Good afternoon, everyone. I hope you are doing well in this time of pandemic. I, Patricio González Faune, is thankful to the organizers of InSyB 20 for providing me this opportunity to speak in front of the renowned speaker from Asia. Well, I am a fifth-year student of biotechnology from Universidad Católica del Maule, Chile. And today I am presenting my final year thesis work, which is entitled as Computational Study on Temperature-Driven Extraction Function Relationship of Biofilm Producing Glycosyl Transferase Enzymes. Okay. To start with today's presentation, I will talk a brief about heat-loving microorganisms. As we all know, microorganisms are present in all the biomes of our planet, which correspond to an immense and very diverse group, and they were the first to inhabit the planet when the Earth was boiling hot. And nowadays, they are linked mostly to hyperthermophiles and archaea. Their job being in just physical-chemical conditions such as hot spring, deep seas, glaciers, sealing lakes, extreme pH, affluent from industrial or mining wastewater, and more. All these microorganisms that are capable of living in these extreme environments are called extremophiles, which generally correspond to archaea and bacteria domains. As we can see in this slide here, heat-loving bacteria mostly comes under the group of archaea. Heat-resistant bacteria can grow at high temperature, ranging between 40 grades and 122 grades Celsius till date. There are three increasing temperature range where we focus to develop this research, which are mesophiles, thermophiles, and hyperthermophiles. An important point to carry out this research was that Chile has a great variety of climatic conditions, which make it a country with great genomic and bacterial richness, from which many bacterial species might represent an economic interest. Another line of interest was due to increasing anthropogenic footprint in the hostile environment due to tourism. There can be risk of emergence of pathogens, and biofilm can be key dominating factor for both. As the images can be seen, we hide the most abiotic point of the Earth, Atacama Desert in the north, with very high temperature, warm temperature in the central area, and cold temperature in Chilean Patagonia, such as Torres del Paine, with very cold climate. To survive under any extreme environment, the common strategy of microorganisms are to form a protective cap of biofilm. The extreme factor can be anything, as we discussed earlier, even antibiotic or drugs. This is one of the reasons why extreme drug resistance occurring. Biofilm forms a cap through which the antibiotic or any other extreme factor like extreme heat or cold do not reach to the microbial colony. Glycosyl transferase group of enzymes are the ones that control the production of biofilm in a way by synthesizing capsular or exopolysaccharide, which is main constitutive part of the biofilm. And here you can see our methodology in brief. The computational study on biofilm producing bacterial glycosyl transferase enzymes from mesophiles, thermophiles, and hyperthermophiles was mainly divided into two steps, a structural and functional study. The structural study consisted of physical-chemical characterization, primary, secondary, and tertiary structure analysis modeling and validation, and while the functional study consisted of protein-protein interaction, membrane topology, motifs and domains identification, binding of enzyme substrate, and molecular docking. Finally, we try to interpret the inner relationship among them. From physical-chemical characterization, high aliphatic index, pi value, gravity, and instability index directly correspond to better structural stability of the acquired enzymes in thermophiles, followed by mesophiles. From the amino acid composition analysis, isoleucine, leucine, and valine, other than glycine and alanine, found common in all the groups, which confirm a stable structural core. Thermophiles were found to be most structurally stable, followed by mesophile. The presence of charge residues in a protein determines the structural plasticity of a protein due to more ion-ion interactions. Interestingly, mesophiles have been the most structurally plastic among all. Folding and folding on alpha helix and random coils directly hints at the formation of high-order tertiary structures. Hyperthermophiles, being more rigid environment of high temperature, found to have the maximum of alpha helix and random coils, followed by thermophiles and mesophiles. The similar results of high-order tertiary structures has been also confirmed from salt bridge analysis, where hyperthermophile consisted of it most, followed by thermophile and then mesophiles. Hydrogen bonds make a large contribution to protein stability by increasing the packing density. When a protein folds, 70% of the peptide group and 65% of the polar side chains are buried in the interior of the protein out of contact with water. Hydrogen bonds plays a major role there. The greater packing density was found in hyperthermophiles. From different structural quality assessment analysis like 3D, 1D percentage, ERAT confidence score, Q-min-C score, or the percentage of amino acid presence in favored region of Ramachandran plot, it was observed that hyperthermophiles and thermophiles were better in results compared to the mesophiles. Here we can see the Q-min results, one from each of the respective group. First, Lactobacillus plantarum mesophiles, second, Rhodothermus marinus thermophiles, and third, Thermus aquaticus hyperthermophiles. For the functional analysis and protein-protein interaction, a string database was used. We can see here one from each respective group. From the left, A, Lactobacillus plantarum mesophile, B, Rhodothermus marinus thermophiles, and C, Thermus aquaticus hyperthermophiles. And repeated interactions were possible to identify among all with different enzymes involved in cell wall formation, polysaccharide synthesis, and ligations. Most commonly known interactions were found in mesophiles, followed by thermophiles and hyperthermophiles. From the membrane topology study, it was observed that a mostly glycosyl transferase is an inner membrane enzyme, while only a few were found transmembrane too. For domain and motif determination, we have not found any significant results, and so we are omitting that part to discuss here. Finally, a molecular docking of the biofilm producing glycosyl transferase was performed with its substrate UDP glucose. Thermophiles have shown better substrate binding ability with most negative binding energy. In conclusion, we may state the probable reason of the less structural plasticity and interaction of the hyperthermophilic glycosyl transferase can be its optimum activation at very high temperature, resulting into a unique rigid configuration from an industrial point of view. Production of the bioactive polysaccharides, thermophilic or mesophilic origin bacteria, might be preferred for more interactive nature of the enzymes, while thermophiles due to its unique structural stability. From a biofilm-mediated infection and pathogenesis point of view, its EPAs producing enzymes like glycosyl transferase might be targeted to attack the biofilm formation. And finally, I pay my acknowledgement to my University UCM Department, School of Biotechnology, for the laboratory CEM and Symbio UCM. I am so thankful to the finding of Fundesit initiation and for the group of international collaborators of the University of Burwan, India. And thank you all for your patient listening.
Thank you, sir. That was informative. Moving on to the next speaker, I now would like to take the pleasure to invite Dr. Renika Suravajla, Senior Research Associate, Manipal University, Rajasthan.
Dr. Renuka is working as Senior Research Associate at Manipal University, Jaipur. She has done her PhD from Roskill University and has 10 years of research experience. She is a recipient of JNCSR Ramdas Pai Fellowship and has worked in IISc Bangalore and IICT Hyderabad. With research experience in synthetic chemistry and cancer biology, she worked on selected drug target related studies with a vivid interest in application of molecular imprinted polymers and also turned herself as a structural biologist with a keen interest in computational prediction studies related to protein ligand, long non-coding RNA related studies. Now, I would like to invite Dr. Renuka to deliver her talk on phytochemical antiviral agents as potential targets for SARS-CoV RNA polymerase. Ma'am, please.
Yeah, thank you very much for this invitation and as well as for giving an opportunity for me to participate in this session. Just a minute. Yeah. Fine. Are you able to visualize my screen?
Yes, ma'am. Yes, it is.
Oh, fine. Yeah. Thank you very much for giving this platform to share my research where I'm working on. And thank you very much for the collaborators as well. And today I'm going to present the recent work which we are doing on phytochemical agents, antiviral agents, which can be used as a probable potential drug targets with respect to the SARS-CoV-2 RNA polymerase. And why RNA polymerase is important for in this study, and why we have to actually target and try to find some potential targets. So in this regard, I would like to thank all the contributors of this work, especially Dr. Jalita ma'am, Hima Kumari, Vidu, Vaishali, Abhinav and all. Yeah, and also Kavkishore sir, who has been the back principal investigator and generating this idea to come forward for doing this work. Yeah. Basically, I'm a chemist, a synthetic chemist turned into a structural biologist, where I have my scope of interest in synthesizing some molecular imprinted polymers, which can be used as a synthetic receptors. And thereon, I have these can be used as biomarkers and developing biomarkers and biosensors. And same as furthermore, to combine both these two basic fundamental studies, I have moved myself into the structural prediction studies, which is nothing but the structural biologist, which can bridge both the things and then be a wonderful chemist. And so I'm looking into these aspects. And today, we have considered due to this pandemic, we are considering SARS-CoV-2 RNA polymerase in this rationally for this work. And the reason behind is SARS-CoV has two types of basic differential proteins, which are nothing but the structural proteins and the non-structural proteins. And as we have a lot of in the market, there are a lot of antiviral drugs which can be used for the treatment, for as a cure, as a remedy. But still there are a lot of adverse effects with respect to these antiviral drugs. So to have a better way to reduce the adverse effects, we have asked that whether some other natural products can be a better solution for these antiviral agents. And we started investigating and finding the ways to digging the thing literature, finding algae, higher plants and mangroves, and all these the sectors where we can find the natural products which have the bioactive compounds. And we in this process about 120 natural products we were able to screen, and then and we were able to use them to know the bioactivity of these compounds on RNA polymerase, which is a non-structural protein of SARS-CoV-2, and the structure the protein structure which we have considered is 7BV2. And why the RNA polymerase is considered is it contains NSP. It is a mixture of different conserved motifs, and it is a domain. It is a protein which is a complex of NSP12, NSP7, and as well as NSP8 as well. And it has almost 7 conserved motifs of them. The A, B, C, D or the PALM subdomains, and as well as the C domain, which is the catalytic active replication domain, and the G and F and G or the finger domains, and which interact with the template of RNA, strand RNA, and it helps in the synthesis as well. And the motif F is present, which binds to the primer strand RNA and also stabilizes the nucleotide. So the further reason that the vaccines and whatever upcoming work is going on is with respect to the RNA polymerase protein only. So we are also very curious to work and dig in this aspect, and because it is a lot of the potential antiviral drugs which are also targeted towards the RNA polymerase. And some of the available antiviral drugs such as remdesivir and rabiferrin, and all these drugs are still in the market, with which have the potential binding affinity towards the RNA polymerase. So we have made a pivotal methodology for this study, and wherein we have screened almost 120 bioactive compounds, and using a target RNA polymerase of SARS-CoV-2, and started working on the molecular docking studies to find the binding affinities and its interacting residues. Furthermore, knowing the stability can be investigated using molecular simulation studies on these screened potential bioactive compounds. And after doing the docking studies, we have come up with some almost 28 potential bioactive compounds, And some of the I'm sharing the names of these compounds which are potential, and furthermore the binding energy and their with respect to all these screened compounds have been I'm sharing with you, but we are looking for publishing it very soon. So, yeah, and these docking studies with respect to sensoside, A is the most potential ligand which we have observed, and it has the similar binding towards the domain A, and these are the various there are various binding with residues which have the hydrogen bonding and with the ligand. So these are some of the efficient binding residues such as the ASP 760 and as well as the ARG55 residue of the RNA polymerase, and as well as the lyase 55, or all these green ones which are having the hydrogen bonding, and other orange one is with the pi bonding. So these the 3D structures have been developed using the chimera and as well as the drug discovery tools. And furthermore, the molecular simulation studies have been done to find the RMSD value of the protein ligand, and as well as how exactly ligand is stabilizing the complex, how it is stabilizing the complex. So these studies have been done by Vidu from IIT Allahabad and using Schrodinger, and the same another potential ligand is psychosapien, which is also having the good potential bioactive compound with respect to the RNA polymerase, and it is also able to form hydrogen bonding with use 760 727 and as well as TYR 717 residues, and this is and 5 alkyl bonding is present with respect to GYL 726 and ASN 722, and so this means these are similar to the residues which are present which are similar to that of the remsidis, where binding residues with respect to RNA polymerase. And some of the similarly, we have also done the molecular simulation studies with respect to psychosapien and the RMSD values and the binding stability or energy has been also studied. Similarly, the same saponerin is also the one another potential ligand bioactive compound which was identified, and with have the binding residues with RNA polymerase, and where various binding residues are also the same with the presently available antiviral drugs with respect to with respect to the COVID, or still this, having the same residues. So we feel that these are some of the potential ligands which have the high good binding affinity, high binding affinity with the RNA polymerase, and the Ki values of the concentration can be very minute, where they can be administered and can be used as antiviral agents. So And in conclusion and future directions with respect to this molecular simulation is we can consider these compounds and go for in vitro studies, and then find how the toxic if they are with respect to the toxic plasma levels and so on. So this is the work which we have carried out, and we are still going looking into the things, and then soon we will be publishing this work. Thank you. And I thank Dr. Professor Kavikishore sir and the organizers of the InSyB 20 and Vignan University team, and as well as UCB Dehradun Anush Kumar and IIT Allahabad team and Dr. Babita Malik from Manipal University, Jaipur, for contributing and being collaborators for this work. Thank you.
Thank you very much, ma'am. That was illuminating. Moving forward, I now would like to present over here the next speaker, Dr. Abhinav Parashar, Assistant Professor, Department of Biotechnology, to take over the session.
Dr. Parashar is an alumnus of VIT Vellore, from where he pursued his MTech in biotechnology, followed by his PhD in enzymology of heme enzymes. He has published a total of 22 research articles with a cumulative impact factor of 67 and has achieved H index of 9. He is an important part of a newly developed mud-burn concept which has brought a paradigm shift in the understanding of redox reactions like drug metabolism, aerobic respiration, photosynthesis, etc. After completing a DSD PhD project as a research associate on developing a diagnostic kit for homocysteine, he is serving as an assistant professor at Vignan University for Science, Technology and Research for more than 3 years. Now I'd like to invite Dr. Abhinav to deliver his talk on curcumin, a potential therapeutic agent against SARS-CoV-2.
Sir, please. Sir, could you please unmute yourself?
Sorry. Okay. Thank you, Akhila and Swana. First and foremost, I would like to thank the team of InSyB for giving me an opportunity to present a recent work that we have suggests. This is a work which is on curcumin, and we have tried to exploit and figure out how good this could be as a potential therapeutic agent against SARS-CoV-2. So, moving further. I guess it's frozen. Okay, fine. Yeah. Am I audible?
Yes, sir.
Okay. I guess my screen has frozen, so I'm unable to exactly see which slide I am on right now. I wanted to talk about the drugs that are currently being used against COVID. There are different types of therapies that are being explored. We all know that there's no drug as such for COVID-19. So the first line of defense, or just to suppress the symptoms, the maximum usage is of acetaminophen, which is paracetamol. And following that, there are antibiotics and bronchodilators because, as Dr. Anil and others were also mentioning, COVID-19 causes a lot of stress on the lungs, on the respiratory system. So to tackle that, we are using a lot of bronchodilators. And hydroxychloroquine was one of the first drugs that was, striked as a potential measure against COVID-19 and was widely explored at that time. But later it came, we all came to know that it's not that effective. Then came plasma that was recovered from the patients, from different patients, and we have been trying to use plasma as a very potential measure against COVID-19. And then there were a lot of antivirals which came up, and some of the major ones were ivermectin and favipiravir, etc. So in total, apart from these drugs, we have also seen that non-conventional drugs such as homeopathy, Ayurvedic, Chinese medications. So all of these are also explored to somehow control the effects of COVID-19. Now, keeping in line of that, we tried to explore one of the magic molecules, curcumin, which has shown great efficacy against so many diseases. So we chose curcumin as our main target, and then we wanted to explore how effective is it, and we wanted to compare it with other currently being utilized drug molecules for treatment of COVID-19. So our methodology, this is the simple representation of our methodology, where we had chosen some of the structural and non-structural proteins from the virus SARS-CoV-2, which was all retrieved from the PDB, then available structures, and then we prepared. After that, we followed the preparation of the molecules and the grid system using Autodoc. We did some docking. It was all an in silico analysis to find out how effectively curcumin will bind to the structures of to the different proteins of COVID SARS-CoV-2, and whether that can be used as an inhibitor for the proliferation. So we used Autodoc. Autodoc uses MGL tools and Autogrid and Autodoc for the preparation and docking purposes. Once that was done, we moved on to visualize the different docs, and and to compare how the locations of the molecules where it is binding, and further for the presentation sake. So for that, we used Pymol and Chimera. Now, a brief, all of us certainly would be knowing the structure, but still, on how we chose the different proteins and where are they located. This is the representation of SARS-CoV-2, where we can see we have an envelope. We have an envelope protein where we can see the spike protein embedded over there, and we also have these membrane proteins. So these serve as the locations which spike protein is the one which identifies the ACE2 on the human cell, and then it finds its way through the skin in our body. So, and a lot of that is present in the lungs. So that's why lungs is the most affected organ. Now, when we go a little inside, we'll see there are nucleocapsids, and nucleocapsid in where you'll find the RNA enclosed. And then we have some other proteins, non-structural proteins like NSP10, 16, and so on. So what we did was we chose the proteins, the important eminent proteins, right from the surface to one that is inside. Once we had selected the proteins, we selected the different molecules that we had to explore, and for that we chose some of the important molecules, some of the important drugs that were currently under use, like azithromycin, which is an antibacterial, and then we chose ivermectin, which is an antiviral, and then we can see that the structure of the molecules that we have chosen here varies a lot. There's a lot of structural differences, and most of them are antibacterial and then antivirals as well. And then we have this molecule, curcumin, which was explored in a little more detail. So moving on to what happened after we did the docking. What did the studies reveal? So we can see on the left-hand side, this is the spike protein, and on the right-hand side, we'll see the membrane proteins. So what we are observing here is, on the end, these are the molecules which showed relatively greater affinities on the spike proteins compared to there were a total of 15 molecules that were explored for this particular study. And we can see that there are something around 6 molecules, including azithromycin and ivermectin. But we can also see that hydroxychloroquine doesn't qualify in this search. So where are the different locations? That is where we were more interested in. So what we see here is N-terminal domain, and then we have the heptad repeat, and then we have the connector domain. So heptad repeat is for there are 2 heptad repeats, and they are associated. They are involved in attaching the spike proteins. So we see that there's only one molecule that found its place in the N-terminal domain, and in heptad repeat and in connector domain mainly, we can see most of the molecules finding a better site to bind. So if the theory goes like this, if the molecule is binding to the connector domain, obviously it is going to affect the binding of spike protein with the this is with the virus, so it is going to affect its presence in the virus itself. And if that is obstructed, then certainly it's going to reduce its efficiency, reduce the efficiency of the virus to attach with the human cell. But we also see that the curcumin, which we are interested in, does not qualify in here. The curcumin actually gave binding energy of something around -5, which is which would have a Ki value of something around 350-60 micromolar. So still not very. It's not a very bad result, bad value, but in here the molecules that are represented are having Ki values of lesser than 50 micromolar. So and that were compared with the Cmax value, that is the amount of drug that will be present in the plasma. So we saw we will see that the other protein, the membrane protein, which is also on the surface of the virus, we see that the membrane protein also did not came up with curcumin as a very good binding molecule on the receptor binding domain or the peptide domain either. So peptidase domain, this is the area where the enzyme breaks the molecule over here, peptide, and then it allows binding of the protein efficiently to the surface of the cell, human cell. So in these 2 protein complexes, we did not find a very good efficiency of curcumin. But then as we moved down inside, we found that nucleocapsid, there is a region here in the nucleocapsid, there's a loop region where we found most of the molecules binding at the same place. So as a very all of these we can see that curcumin also binds to the same location, and with a very high binding efficiency, which is something which is lesser than 2 micromolar. So if curcumin is able to bind with almost equal efficiency like ivermectin and azithromycin, that is what we had observed over here, lesser than 2 micromolar for ivermectin and azithromycin as well. So curcumin qualifies equally strong a drug candidate or a molecule that can be used against SARS-CoV-2 for the nucleocapsid. Nucleocapsid is the protein that attaches the RNA of the virus. So if this is obstructed, if the structure of nucleic acid is obstructed, then obviously the binding of RNA to this will be obstructed. Now, yeah, and then there's another molecule which is NSP10. NSP10 is in complexation with NSP16, and it recognizes the RNA molecule. It helps RNA molecule while for the proliferation part, and we can see that there are zinc fingers around here. Zinc fingers are the ones that will allow NSP10 to identify RNA molecule, and we see that these drugs, wherein we have curcumin as well, again azithromycin and ivermectin, these molecules are binding exactly in the zinc finger groove. So once this happens, once these molecules reach out there in the zinc finger groove, obviously the binding of NSP10 to RNA will be highly improbable. So we saw this is some of the preliminary results that we had got, and what we have observed from this is that curcumin showed a significant impact on nucleocapsid and NSP10, these two proteins of SARS-CoV-2, and if not standalone, then obviously at least curcumin can definitely be used as a combinatorial drug for the administration against SARS-CoV-2. And a little more study in this, probably some in silico and the in silico study is done, probably some in vitro study and in vivo study will reveal the regulating activity of nucleocapsid and NSP10 proteins and how it interacts with curcumin. So this is the end of my slide. Thank you for your patient listening. I request any queries if there are. Thank you all.
Thank you, sir. That was clear-sighted. I now would like to request Dr. S. Anil Kumar to give concluding remarks for this day-long informative session. Sir, please.
Thank you, Suhana. Dear all, thanks for participating in this two-day event. And it's the time to close the day one session, wherein we had very good lectures by eminent speakers across the globe. And the session started with the inaugural function by our Honorable Chairman, Dr. Laura Rathaya, followed by inaugural session by our Vice Chancellor, Dr. M.Y.S. Prasad. Later, our Chancellor released Book of Abstracts. Then our registrar has talked about our university, followed by Dr. N. Jaleja giving insights into InSyB and APBionet. Then Professor Kupanidhi sir has explained about the Department of Biotechnology, and Dr. Rangarao has explained about the genesis of InSyB. And now the day has completed with two technical sessions and followed one inaugural session followed by two technical sessions. In the technical session one, Dr. Jagdish has introduced Dr. Kenta Nakai, who is expert in NGS data, followed by Dr. Narahari Shastri, who has been the invited speaker for session one. He has delivered very beautiful things on his topic, understanding relationship between infection and immunity. Then Dr. Bruno Guetta, Dr. Ramaswamy has delivered their lectures, followed by Ramaswamy, Dr. Keith Yam. Madam has delivered on structural bioinformatics in drug discovery. Later, young scientists or young guys like Anil Kumar, Fane, Renuka Surojala, and Dr. Abhinav Parashar has delivered their lectures. Now it's time to close the day one session, and we will meet tomorrow at 9 a.m. sharp. And at the same time, I would like to say one more thing. That is, after the concluding of tomorrow's session, I will send the feedback link. After filling that one, you will receive your certificate copy. Thank you for participating, and very thanks to all invited and keynote speakers for delivering a wonderful lectures and giving insights into the various topics and which would have ignited our minds. Thank you all. Signing off until we meet tomorrow. Thank you. Thank you, Suhana.
Thank you, sir.
Sir.